This conference will now be recorded.
Hello everyone. Well, since I hope everyone is here and we have a lot to cover, we really should begin today's conference. I really appreciate you all for attending today. A very good morning, afternoon, or evening to one and all present here virtually with me. Let me just introduce myself. My name is Rachel McLean, Conference Secretary at Coalesce Research Group LLC, and I will be available for any of your queries or questions throughout the conference to help you out technically and organize the conference successfully. For the next 5 minutes, please allow me to give brief description about our organization and also further instructions. College Research Group LLC is very delighted to welcome you all the speakers, supporting association students, and delegates present here virtually with us today. With the aim, mark the excellency by facilitating conversations, sharing knowledge, College Research Group follows throughout the objective of setting standards in delivering excellency in the research among the researchers, academics, and industries. Procumulating the research throughout conducting events on online webinars from various fields of science, especially from the stream of medicine, science, engineering, and technology. Within a short duration, we have succeeded in creating a platform for developing a strong professional network with the people across the world from their respective fields, and they haven't connected in a while. As a global scientific event organizer, we plan and carry out international conferences, meetings, seminars, symposiums, and workshops providing the best delegate experience, excellent networking opportunities, conversations leads to innovations, learning new opportunities, lasting work relationships, and professionalization with the professionals around the global network. Just recently, we have also started our own platform for the open access pre-reviewed scholarly literature, where all the content is freely available without any charge to the readers. We aim to share the new recent advances throughout the scientific publications, which helps in the minimizing the borders and contributing the use of research work of eminent authors, scientists, researchers, research scholars, students, etc., across a broad spectrum of medical, clinical, pharma, and engineering scientific disciplines and its sub-disciplines to promote the ideas which will guide the development and facilitate the use of scientific informations and ensure its quality, and also will be connecting link between the researchers and scientific community. We are committed in ensuring that our journals are disseminated worldwide so that the researchers are able to access our content wherever and whenever they need it. As being very new journal, we have coined up some interesting journals and have also formed the editorial boards, and we will be very glad if any of today's attendees, speakers, students, or delegates could extend your support by checking the journal, and will be interested in publishing an inaugural issue at any of the journals according to your area of expertise. We will also send an email with all the details about the journal after the commitment of the conference. Do take your time to check the website. Thank you. Finally, I would like to thank all the attendees and friends and gain knowledge from sessions today. We also hope your participation will contribute to the new research findings and that will lead to the new collaborations and research intakes. Before we start with the sessions, I guess some of you will be the new to GoToMeetings app. Let me just explain you the important tools required for the presentation. Before we start with the sessions today, I guess some of you will be very new to the GoToMeetings app. Let me just explain you the important tools required for the presentations. On the GoToMeetings app or web page, you can find four icons at the bottom center. One is for mic, one is for camera, one is for screen, and one is for end button. Mic button. Click on the mic button to mute or unmute yourself. I request all you to stay muted while our speakers are presenting the research work so that it does not cause any disturbance to them. Camera button, you can use this button to share or unshare the web camera. If you have the option to share the webcam, you can do it and it's completely your choice. Screen button, you can use this button to share or unshare. This is the main part during your presentation after I call your name from my side, and I will be making you as a presenter, and only I'll give the permission to share the screen, then only you will be able to share your screen. So once I make you the presenter, you can click on the screen button and you will find a pop-up window to share your screen. End or leave button. This is your fourth icon where you can use this to leave the sessions. On the top center, you can use the options to change your display settings. At the top right corner, you'll have three icons: people, chat, and settings. In the chat option, you can chat with me or any other participants. For the settings, you can use this button to check for the settings of your speakers and microphones. As I mentioned, kindly remain muted during the presentation so that the presenters do not get disturbed, and further any questions, you can ask the questions or you can send us your questions in the message box too. Well, let's not to take much of your time. Finally, I would like to request you all to please give us your brief introduction about yourself before starting with the sessions so that we can know about you virtually. And now let's move into our today's session. Our first presenter is Beneditti Fromiger from Uni Montpellier, France. Doctor, are you ready with your presentation?
Yes, I'm ready.
Okay, now I'm going to make you as a presenter now. Please share your screen.
Yes. You can see my screen?
Yes, we can see your presentation.
Okay, perfect. So I can begin?
Yes, you can continue with your talk. Thank you.
Okay. So hello, everybody. Thank you for the committee to let me present my work. So my name is Bénédicte Fromager. I'm a second year PhD student at l'IEM, Institut Européen des Membranes, at Montpellier in France, under the supervision of Julien Cambedouzou and David Cornet. So I'm working on 3D scaffolds of pan-electrospun fibers for cell culture. So I will first present, do a brief introduction on 3D scaffolds. Then I will present my scaffold, the main characteristics, and how I produce my scaffold. Then I will present some topological characterization, then the mechanical characterization, and finally, the perspective I have. So first, why do we need cell culture scaffold? So when we need to study disease evolution, for example, or pathway, we need to study cell. So we have to do cell culture. Also, to study the impact of drug, we are first going to study it on cells. Also to do cell therapy, which so we have to seed stem cells and induce their differentiation into specific cells that are used then to regenerate defected tissues or organs, for example. Also to do personalized medicine, which is to find the best treatment for each patient, because we have to perform genetic, proteomic, and metabolomic studies. We study also biomarkers on cells that are seeded on cell culture scaffold. And finally, we can do bioproduction. The bioproduction, I remind, is production of biological molecules that are useful for cell therapy, for gene therapy, production of vaccines, and, for example, to cure some difficult disease. And to study disease pathways and impact of drugs, there are several steps. So what for? First, we have to study the disease evolution or the impact of drug on cells. When it's okay, we study it on animals. And finally, we test it on human. However, when we study it on animal, it takes time and money, and it presents some ethical issues. So the objective is to produce a cell culture scaffold mimicking at best the extracellular membrane matrix, so that we would be able to pass directly from cells to human. So now, why 3D scaffolds? When we seed cells on 2D scaffolds, we have observed that the cells lose their differentiation and present some neoplastic cells properties. When we seed cells on 3D scaffolds, what is good is that it better mimics extracellular matrix that has 3D physical cues. Also, it provides biologically mimetic model systems to study pathways, differentiation, metabolism, proliferation, migration, as well as a scaffold for tissues engineering and regenerative medicine. When we will be able to have 3D scaffolds mimicking extracellular matrix at best, we will be able to reduce use of animals, and at the end, to prevent use animals. And it will also help us to save time and money, as I said before. So there are different kinds of 3D scaffolds for cell culture. So first we have multi-layer cell culture. So the advantages are that they are composed of matrix extracellular, also growth factors and some tissues. However, it's hardly reproducible and it's really, really long. There are also hydrogel. So they have some advantages. Usually we use highly hydrated polymers to produce hydrogel, but they present limitations. For example, when we use natural polymers, it's difficult to have some reproducibility. If we use, for example, highly reticulated polymers, some of them are really cytotoxic. Then we have sponges. However, it's not easy to monitor the rigidity, the porosity, and degradation rate. We have fibers. We have also organoids that are multi-cell phenotype structures that are self-organized. So they are really good to mimic the extracellular matrix. However, it's hard to have reproducibility, and also it's long as multi-layer cell culture. And finally, we have 3D printed scaffolds, which the advantage is that the computer helps to control specifically each parameter. However, cells bear stress and pressure if we bioprint them. So the fibers. My scaffold is made of fibers, so I'm going to now present my scaffold. So, which is made of pan-electrospoon fibers. So it's a low-cost routine use, it's reproducible and synthetic. The fibers present fluorescence properties that help us to study migration. It's biocompatible and stable, so there are no degradations that would impact the results, and we can sterilize it for cell culture. We can perform different studies, so omic studies, for example, because the scaffold will not degrade itself, so we will only get the proteins that are the biomolecules that comes from cells. We can perform mechanical, chemical, and physical properties independently. We can study differentiation, metabolism, proliferation, and migration. This scaffold can be used for tissue engineering and regenerative medicine since it can be functionalized. And finally, the first studies showed that it mimics the in vivo, and so it improves cell assays and the prediction accuracy. So how do I produce my scaffold? First, I have to prepare the solution that is made of polyacrylonitrile that you can see on the top right corner of the screen in DMF. I mix it at 70 degrees Celsius. And sometimes for some of my scaffold, I add a multi-wall carbon nanotube. And you can see here the 4 different amounts of carbon nanotubes that I add. So 0%, 0.0015%, 0.00625%, or 0.05%. So why do I use PAN? Because first, it's a best precursor for carbon fibers formation because it has a great carbon yield and a high melting point. It presents really good physical and mechanical characteristics. It can be used in biology because it's biocompatible. We can sterilize it, and it shows resistance to biodegradation, as I said before. We can design and act on mechanical properties and physical chemical properties independently one from another. And the PAN is soluble in low toxic solvents. So when the solution is ready, I electrospin the solution. So at my laboratory. So I put a solution in a syringe on a syringe pump. The solution reaches the needle tip, and we apply a high voltage difference between the needle tip and the rotating drum, that is a collector. So we have the creation of a field between the two, the needle tip and the rotating drum. And so at first we have the formation of a droplet at the needle tip. So we have surface tension. And by increasing the voltage, some electrical forces deform the droplet, and we have the formation of Taylor cone. And above the critical voltage, the jet forms itself, and then it gets thinner, stretches, and it solidifies. So during the jet travel towards the needle tip to the collector, the solvent evaporates. So when they reach the collector, we have solid fibers. So different parameters must be taken into account for the electrospinning. So first, the voltage, that is usually between 10 to 30 kilovolts. I use 20 because, for example, by increasing voltage, we decreases diameter of fibers. We can also act on the flow rate, that is usually between 0.1 and 4.5 milliliter per hour. So by increasing the flow rate, we increase the production. However, beyond a critical flow rate, we produce beads fibers. We also can act on distance between metallic collector and the needle tip, which is usually between 5 centimeter and 20 centimeter. So this depends on the evaporation rates of the solvent. Then we can act on the needle diameter, because by increasing needle diameter, we increase diameter of fibers. We act also on polymer concentration, because by increasing the concentration, we increase the formation of defect-free fibers. Also the conductivity, we can act it on adding some salt, for example. When we increase the conductivity, we increase the charges on surface. so we decrease the diameter of fibers that are formed. And finally, the choice of the solvent is really important. First, the polymer must be completely soluble in it, and it must present moderate boiling point because if it's too volatile, the solidification will be too quick and the fibers will not reach the collector. But if it's too low, we will see bead formation and some wet spots because the solvent will not have time to evaporate. So after having electrospoon it, I can act on the rotation speed of the collector to obtain either aligned fibers or non-aligned fibers, as you can see here. So when I have my fibers, I apply a heat treatment. So I from 0 degrees to 250 degrees. So during this heat treatment, the polyacrylonitrile overcome a cyclization and a dehydrogenation and an oxidation to form a ladder-like structure. So this enables to have a structure that is infusible and reticulated. So it enables us to sterilize the membrane after that. And also the function presented allow us to seed cells that can interact with the fibers, and also we can make functionalization. So here you can see the IR spectrum of the nanofibers before in red and after in blue heat treatment. So after that, I obtain fibers that present a yellow color, and I characterize it, I can functionalize it, and perform cell culture. So as for now, I show you that I produced a 3D scaffold for cell culture. This scaffold is obtained from electrospinning of a span solution. The stabilization of the scaffold by heat treatment make it possible to sterilize it and use it for cell culture. So now I'm going to present some topological characterization. So first, I take 3 scanning electron micrographs for each sample that are mounted onto same stubs with carbon tape. Here you can see on the bottom left corner of the screen the membranes that I put on the transversal way. And with the scanning electron micrograph, I can see that I have aligned fibers. And on the top right corner, here it's when it's on the surface. And here you can see the fibers also. So when I obtain the same images, I used a plugin called Diameter G in Image G to have a lot of information. So this plugin is a two-step plugin. So first, the image is segmented. So it produces a binary image with the fibers in white and the background in black. And during the second step, we have some algorithms that enable us to obtain information on the diameter of fibers, on the orientation, and on the area of pore. So here you can see for the first step of the segmentation, it uses 16 algorithms. So we obtain different kind of segmented images, and I choose the one that you can see in the circle because it's the most representative of my scaffolds. So from this topological characterization, what I could see is that, so first I took one membrane at different places and I performed studies. And what I could say is that I must cut the edges because they are not they present some different characteristic. Then I compared different membrane of the same solution, so with only pan, for example, or pan with one amount of carbon nanotube. And what I could see is that it's reproducible. And also I compared without and with carbon nanotube, and I could see that the addition of carbon nanotube doesn't impact organization. So here you can see some of the results I obtained. So you have both aligned and non-aligned fibers with and without carbon nanotube in the first solution. And what I could say is that I produce nanofibers of pan of 0.681 micrometer, and the pores are around 27 micrometer per square. So then I tried to modify the pore area. So what I did is that I took the fibers before the heat treatment. I hung them from one side on the top, and on the other side, I put some lead weights, so either perpendicularly or parallel to the fiber orientation, as you can see on the screen. And you can see that when it's perpendicular to the orientation of fibers, I obtain that structure with waves, whereas when it's parallel to the orientation, I increase the orientation of the fibers. So here you can see better. It's so first I hang without any lead weights. Then the second one are perpendicular and then parallelly to the orientation of fibers. So to summarize this part, I used a plugin called Diameter G to analyze same images, which helped me to say that my nanofibers have a diameter of 0.681 micrometer and pores of 27 micrometer per square. And when I apply traction during heat treatment, I either deform or increase the alignment of fibers. So now I will present mechanical characterization. For that, I used atomic force microscopy in Bordeaux, and I study the fiber alone, which present, as you can see here, gigapascal modulus, and fibers within in the membrane, and they show that I could show that it's around megapascal, the modulus. Then I wanted to see the organization of the carbon nanotube within the membrane. So I use TEM analysis. So first I had to perform ultramicrotome cut and analyze it with TEM. And here you can see two different things. So on the left, the pan nanofiber without carbon nanotube, and on the left with carbon nanotube. So what I could say from the image obtained of the membrane with only pan is that I can see that the fibers are really circle one. So the fibers were really aligned when I performed the cut. And on the right, what I can see is that the carbon nanotube are within the fiber. So they are not next to the fibers, but in the fibers. And before putting the carbon nanotube in the solution, I asked for the producer to functionalize it with iron. And I can see that it was good to have the functionalization so that I can really see the carbon nanotube. So then I wanted to see the impact on cells, the fact that I added some carbon nanotube. So in Bordeaux, first formed the neurospheres, then I deposited on fibers, then I fixed them on lame, and finally I studied with fluorescence. And what could be seen with this study is that the cell migrates toward fibrous orientation, and we have seen that there were a migratory capacity maximum for the membrane with produced with a solution containing 0.0015% carbon nanotube. So when I add the carbon nanotube, I have an impact on cells. And what is not showed here is that when I performed nanoindentation, when a colleague performed nanoindentation on my membranes, they could show that there were a difference of stiffness of fibers when I added carbon nanotube. So cells must feel the stiffness, and this has an impact on the migratory capacity. So the fibers, when I perform AFM, I can see that the fibers presents gigapascal modulus, and when they are in the membranes, it's megapascal. The carbon nanotube are in the fibers, and the addition of this carbon nanotube has an impact on the migration of cells. So what are my perspectives? So first, I would like to see the impacts of the modification of the membrane by diffraction during each treatment on cells. I would like to perform electrochemical characterization of my membranes to see if the addition of carbon nanotube has an impact on electrochemical properties. And finally, I would like to functionalize my fibers. So first by adding NH2 functions on the pad membranes, then add the glutyraldehyde, and finally some RGD motifs. And during at each step, I would like to study the impact on cell culture to see if it has an impact. Yes. So thank you for your attention. And if you have any question, I would be pleased to answer.
Thank you, doctor. Is anyone having any questions?
Hi, can you hear me?
Yes.
Yes.
Hi, I'm Claudia Colussi. I have a question. I'm very interested about the growth and migration of neurospheres on this matrix scaffold. And I want to know if there is a modification of neurosphere growth or survival when they are on this type of scaffold.
Yes, it was shown not by me but with the previous student that their differentiation and growth are more similar to the in vivo when they are in 3D scaffolds than when they are on 2D scaffolds.
Okay, thanks.
You're welcome.
Any more questions?
Hello.
Hello.
Yes,
Yeah,
doctor.
do you hear? Yeah, do you hear me?
Yes. Yes, we
Yeah.
can hear you.
I have a question regarding to the mechanical properties you measure.
Okay.
You mentioned that you use the micro CT.
Dr. Ali, like, dude, I
The
think you're having some network
question,
issue.
why you don't use the universal mechanical testing to measuring the tensile properties rather than micro CT?
Sorry, I couldn't understand your question because there were some problems with your connection, I think.
Doctor, Yes. Dr. Ali, I think you are having some
Yeah, do you hear
network
me now?
issue. Can you please? Yes, we can hear you. Can you please repeat your question?
Yeah, the question is regarding to the mechanical properties you measured. You said you have used the micro CT to
Yes.
measuring the mechanical characterization. The question, why you didn't use the universal mechanical testing to measuring the tensile testing, which is more accurate than the micro CT?
The microcity was used to see the organization of carbon nanotubes. So I wanted to see if they were in the fibers or, for example, next to the fibers. And I used atomic force microscopy and nanoindentation to characterize the mechanical properties.
Got it. All right. Okay. Yeah, that's much clear. Yeah, when you use the FM, yeah, got it. Thank you so much.
You're welcome.
Thank you, Dr. Ali. Any more questions? I think there are no questions. Thank you, Dr. Promeger, once again for your presence and presentation. Let's move to our next presentation by Dr. Claudia Coalesce from Antonio Robotai, CNR, Italy. Doctor, are you ready with your presentation?
Yes, thanks.
Okay, now I'm going to make you as a presenter now. Please share your screen.
Okay. Can you see my presentation?
Yes, we can see your presentation.
Okay,
Please
so
continue with your talk.
Okay, thank you. So, hello to everybody. I'm glad to be here and share with you my recent data regarding the role of the nucleoporin 153 as potential target to ameliorate neurogenesis in Alzheimer's disease animal model. I'm a researcher from the National Research Council in Italy. So, Alzheimer's disease is a neurodegenerative disorder characterized by progressive memory loss and cognitive dysfunction that has both a genetic and a sporadic form. The genetic form is a rare disease with only 5% of incidence in the population, and the sporadic form is the vast majority of AD and the main cause of dementia in the elderly. In both cases, we have the accumulation of toxic proteins, which are the amyloid beta peptide and the phosphorylated tau. In the brain of Alzheimer's patients, we have the loss of neuronal cells and synapses, and this occurs in several areas of the brain and also in the ventricles and the hippocampus, which are the main neurogenic niches of the adult brain. These neural progenitors which resides in this zone are important for the processes of learning and memory and for hippocampal plasticity, and they also provide a source for the tissue homeostasis and the response to the damage. However, sorry, can we go? Okay. I'm sorry, the presentation is blocked. Oops, okay. So neurogenesis declines during aging and in several neurodegenerative disorders, including Alzheimer's disease. So we have an impaired neurogenesis that contributes to cognitive dysfunction in Alzheimer's disease, although the mechanism is actually still unclear. Sorry, problem with this. Okay. Okay. However, the mechanism implicated in this defective neurogenesis are still largely unclear, and now the research is focused on the finding of new potential modulators of neurogenesis in order to boost brain repair capacity. So, why does it work? Okay. Okay, in the last years, I have been studying the function of new nucleoporins, which are nuclear pore component and form a large channel across the nuclear envelope and are important for the transport of several molecules, including RNAs and proteins. However, these proteins, especially in the nucleoplasmic part, are bound and linked to the chromatin and are important in the regulation of gene expression, chromatin structure, and genome stability. In the last years, I have characterized specifically the role of the nucleoporin 153 that associate with chromatin and regulates gene expression in several contexts, including the gene expression in the dystrophic MDX hearts, and also contributes to the altered gene expression in prostate cancer. And more recently, a paper from the group of Fred Gage came out about the role of NAPA53 in the maintenance of the stemness of neural progenitor cells, thanks to their interaction with SOX2, which is an important transcription factor for these cells. So in this talk, I'm going to show you the contribution of defective NAPA53 to neurogenesis in AD. I'm also the results for targeting the NAPA53 to improve neurogenesis in AD, and then which are the pathways regulated by this protein. So as a model, we have used the triple transgenic mice that harbor a mutation in amyloid precursor protein, presenilin 1, presenilin 2, and accumulate A beta and tau. So first of all, we checked the NAPA53 levels in the hippocampal neurogenic niche. So I monolabeled this tissue with NAPA53 and SOX2, and I observed that these cells are present in this niche, but are reduced in the hippocampal niche from AD mice. I also found that this defective expression of NAPA53 is maintained in the neurospheres that are formed from newborn mice from this AD model. I also observed that the interaction between NAPA53 and SOX2 that is present in wild-type neurostem cells is reduced in neurostem cells coming from AD mice, as demonstrated by co-immunoprecipitation and confocal analysis. So the first question that we wanted to answer is this reduction of NAPA53 dependent on A beta levels? So we treated the wild-type neurostem cells with A beta for 48 hours and found that the level of this protein was reduced. The level of this protein were also dependent on the nitroxidative stress. Indeed, the treatment of wild-type cells with the ethanol, which is a nitric oxide donor, thus mimicking condition of nitroxidative stress, led to the decrease of the protein. On the contrary, treatment of the cells with L-name, which is a blocker of nitric oxide synthesis, led to the increase of the protein. So from this data, we know that A beta and nitroxidative stress can be important for the reduction of this protein. So we treated AD neurostem cells with the gamma secretase inhibitor to block the accumulation of A beta, and with the aim to reduce nitroxidative stress. So in this condition, we got the re-expression of the protein. Similar results were obtained when these cells were treated with two antioxidant compounds, the N-acetylcysteine and the ascorbic acid, that led to the re-expression of the protein. So we wanted to address the role of NAPO53 re-expression in these cells and see whether this could have an effect on the proliferation of these cells. So NAPO53 was re-expressed in AD cells and confirmed by Western blot and RNA expression. And then we tested the proliferation of these cells, measuring the phosphorylation of serine 10 on histone H3, which is a mark associated to proliferation. In wild-type cells, we observed an increase of a high level of this marker that is reduced in AD neurostem cells and was partially recovered by the overexpression of NAPO53. Proliferation was also assessed by BRDU incorporation, and when NAPO53 was overexpressed both in wild-type cells or in AD cells, we got an increased BRDU incorporation as index of proliferation. We also performed the neurosphere assay, and when NAPO53 was overexpressed, we observed an increased number of neurosphere, and these neurosphere were also bigger. Since migration is an important feature of neurostem cells, we perform an assay to test their ability to migrate and to feel a scratch applied on a plate. So wild-type cells migrate fastly to close this gap, but the AD neurostem cells were much slower. However, in the presence of NAPO53, we had the recovery of the migration of these cells. Accordingly, the expression of the PSA and CAM, which is a modified form of an adhesion molecule important for migration, was highly expressed in wild-type neurostem cells, low in AD neurostem cells, and recovered when the cells overexpressed NAPO53. We also analyzed the expression of two genes important for proliferation and the epigenetic regulation, which are cyclin D1 and TLX. These genes were expressed in wild-type cells, were low in AD neurostem cells, and recovered in the expression when NAPO53 was overexpressed. So the next step was to address the role of NAPO53 overexpression on differentiation of these genes. So we tested the expression of the genes involved in fate determination, that were REST, MASH1, and NeuroD1, and found that when NAPO53 was overexpressed, we observed the expression of these genes that was higher compared to the untreated cells. Differentiation was also assessed by the analysis of the expression of several important markers of early differentiation, such as the double coating, and these cells expressing NAPO53 could express more double coating at day 3, while at day 6 we observed a higher number of beta-3 tubulin positive cells, expressing also the calcium channel CAF1.2, and also at day 10 we observed that these cells show longer dendrites, and where there was a higher number of MAP2 positive cells. Finally, we tested the maturation of these cells, measuring the sodium current that was higher in cells expressing NAPO53 compared to the untreated cells. In the second part of this talk, I want to show you the data regarding the in vivo approach to test and to validate this target for the neurogenesis in this AD model. So we injected in the hippocampal niche lentiviral vector expressing NaPo53. After recovery of 7 days, we injected BRDU for 5 consecutive days, and then we analyzed by histology the neurogenesis, and after 1 month, again histology and the cognitive test. First of all, we checked the expression on NaPo53 in the neurogenic niche. We have GFP positive cells that were also BRDU positive, indicating cells that are going to proliferation. And then we analyzed the early differentiation process by measuring BRDU and double coating positive cells, single and double positive. In the wild type hippocampus, we have a high number of BRDU positive cells that were also double coating positive. This number was low in AD mice according to defective neurogenesis, but this process was recovered in AD mice expressing NaPo53. We measure also the double positive cells for BRDU and PSA and CAM, as we did in vitro, and found that the expression of PSCAM and BRDU positive cells that was defective in AD mice was recovered in mice expressing NaPo53. At 1 month after lentiviral delivery, we tried to see whether there was an incorporation of these cells in the granule layer, indicating their maturation and integration in this part. And we did this by measuring the BRDU double positive cells for neuronal N, which is a late maturation marker. In wild-type cells, in wild-type hippocampus, we observed the presence of these cells that was, however, low in AD mouse, but it was recovered by the presence of NaPo53, in which we could observe the presence of these double positive cells. At this time point, we performed a behavioral test, specifically the Morris Water Maze test. In this test, mice are trained to find a platform in a swimming pool, and in the test probe, they have to remember where the platform is located since it is hidden. Only the mice injected with the vector coding for NaPo53 were able to discriminate the quadrant in which the target was located, and so showing an increase of the memory performance. So finally, we try to find new pathways associated with this ability of NaPo53 to improve neurogenesis, and we immunoprecipitated the NaPo53 from extracts of wild type and the neurostem cells, and then perform a proteomic approach to study the interactome of NaPo53. We found several proteins, common proteins bound to both wild type and AD neurostem cells, but also specific proteins bound only in the wild type cells or in AD cells. So we performed a gene ontology analysis of biological processes in wild type cells and found that the enriched processes were actually RNA biogenesis, processing and transport, of course the nuclear import-export, and then gene regulation, chromatin remodeling, and mitochondrial regulation. Regarding the neurostem cells from the AD mice, these processes were enriched in RNA catabolic processes, energy metabolism, oxidative stress, and proteasomal pathways, and gene regulation. To further analyze these processes, we applied the KEGG database pathway and to find the relevant pathways implicated and relevant for AD. So what we have found is that actually there was an enrichment of pathways common to neurodegeneration and Alzheimer's disease, oxidative stress, mitochondrial dysfunction, and protosomal and catabolic processes. So according to this data, as a proof of concept, we tried to understand whether NAPO53 overexpression could counteract oxidative stress and protosomal dependent protein degradation. So the cells were treated with the DHE, which is a red fluorescent probe for oxidative stress, and cells were labeled with this probe. And as you can see here, we got a low signal in wild-type neurostem cells, a high signal in AD GFP cells, and a decrease of oxidative stress in cells expressing NAPO53. Similarly, we measured the proteasome activity and found that this activity was high in wild-type cells, lower in D neurostem cells, also in agreement with literature data showing the accumulation of toxic and misfolded proteins in neurodegenerative diseases, and a recovery of this activity in AD cells expressing NAPO53. So in conclusion, we have seen that the levels of NAPO53 are reduced in AD neurostem cells in vivo and in vitro. The interaction between NAPO53 and the transcription factor SOX2 here is reduced in these cells and can be responsible for the altered expression of several genes in this context. However, NAPO53 restoration recovers the appropriate response of these cells to both proliferation and differentiation in vitro and in vivo. And NAPO53 delivery in vivo also improves the memory in AD mice. And finally, by a proteomic-based approach, I have shown that there is a NAPO53 protein network which is implicated in the protection from the neurodegenerative, oxidative, and degradative pathways. So this data suggests that actually NAPO53 could be a therapeutic target to boost endogenous neurogenesis. And I want to finish by thanking Alzheimer's Association for funding this project, my collaborators Alessia Bertozzi and Federica Conte, and the group of Professor Grassi at Catholic University for the great collaboration. Thank you to you all. So. Hello?
Hello, everyone, can you hear me? Hello.
Yes.
Okay. Oh, is anyone having any questions? I think there are no questions. Thank you, Doctor, once again for your presence and presentation. Let's move to our next presentation by Dr. Sara Lupis Fendior from University of Nova de Lisboa, Portugal. Doctor, are you ready with your presentation? Yeah, now I'm going to make you as a presenter now. Please share your screen.
Okay.
Hmm?
You can see now?
Yes, I can see your presentation.
Sorry. Because I only see the program for the conference. I don't see my presentation. Mm,
We can see your presentation. Please continue with your talk. We can see your presentation, Doctor. Please continue with your talk.
uh, there is, wait, because there is an error message. Okay, wait. Okay, you see my presentation?
Yes, we can
Yes,
see.
we can see your presentation. Please continue with your talk.
Okay. Good morning to everyone. I'm Sara Ventura, and I'm going to present a project entitled 3D in vitro modeling of cutaneous melanoma for therapeutic studies, and I will start with a brief introduction. Skin, you see the slides changing? I passed my slides, but I don't know if you can see. Okay, I will continue. Skin is the biggest organ of human body, working as the first line of oh my god. As the first line of defense of the external components, and its main function are protection, thermoregulation, and sensations. Human skin is composed of two main layers, dermis and epidermis. The dermis is mainly composed of fibroblasts.
that produce the extracellular matrix, and the epidermis has a thickness that varies according to the body location and is composed mainly of keratinocytes and melanocytes. Keratinocytes form four different layers within the epidermis, being a self-renewal system where keratinocytes are constantly forming in basal layer and corneocytes are always flanking in the upper layers. Melanocytes are known to interact with keratinocytes to regulate the pigmentation of the skin, and when melanocytes suffer mutations and transform into melanoma cells, they invade the basement membrane and proliferate into the dermis. And from this proliferation, melanoma can be characterized according to progression stages. This stage of melanoma is related to the thickness of the tumor, the ulceration, and then spread to lymph nodes or other parts of the body. In the early stages, tumor cells proliferate radially into the epidermis, but over time, melanoma cells acquire an invasive phenotype, breaking the basal lamina to infiltrate the dermis with a subsequent vertical growth. Cutaneous melanoma is the deadliest. Okay, I see your message, Claudia. And yes, this is what I asked when I start, because I don't know, I passed my slides, but I don't know if you don't see. Maybe there is a, but when I pass my slides, you don't see. I just pass my slides now, but you don't see, right? Okay, in this way you can see my slides changing. Right? Okay, maybe I can do the presentation like this. I don't know if I should start in the first slide, or maybe I will continue. Where I'm going. Here, I think Okay. Cutaneous melanoma is the deadliest form of skin cancer and presents a high metastatic potential with increasing incidence worldwide. And it is estimated that there will be an increase of 62% of cases and 74 of deaths in 2040. So why do we want to study melanoma? For this, despite the evolution of therapy for melanoma, there are many patients who continue to die due to the resistance of these therapies. So to simplify, I'm going to refer to the cutaneous melanoma only for melanoma. So how can we study melanoma? One of the issues with the cancer therapy development is the differences between results observed in animal models and in patients, because most models do not recreate the microenvironments of human melanoma, and in two-dimensional cultures, the lack of complexity of interactions between different types of cells does not reflect the physiological human conditions. So over the last decades, there has been a strong investment in development of in vitro 3D melanoma models to study the aggressiveness of melanoma. They are inexpensive and less time consuming than animal testing, and in addition, 3D cultures are more complex systems than monolayer cell cultures, miming the environment more closely, and they don't have the ethical issue limitations of animal models. Since 2000, 3D models of melanoma have been developed. However, they have several limitations. They use collagen from animal origin, don't have melanocytes, and take a long time to be fully formed, for example, 49 days. Previously, in our lab, we developed an in vitro skin model using 3 primary human cells: fibroblasts, melanocytes, and keratinocytes. And in our model, we obtained a good stratification of the epidermis, as well as a good development of the dermis. Melanocytes were aligned in basal lamina, as in human skin, and our model was developed in 24 days. With the development of this skin model, we wanted to know if it would be possible to develop this model with melanoma. So here we can see the protocol that we use to obtain our 3D model. Firstly, we seeded fibroblasts to develop the dermis onto a scaffold. And after this, the other different type of cells were added: melanocytes, keratinocytes, and melanoma cells. And we use 3 different melanoma cell lines: A375, G361, and SKML1. And to introduce these melanoma cells in this model, we tested 2 methods with different concentrations of melanoma cells. One of them is the model CD, where we seeded melanoma cells directly on dermis. And the other method is the model CA, where we seeded cells in aggregates that were previously formed. And after 25 days, the objective is to obtain a human skin with dermis, like you see here, with the dermis and epidermis with the tumor formation. Relatively to the results, in our model, melanoma cells remain in their original microenvironments, and after proliferation occurs the invasion of the basement membrane. Here we can see the hematoxylin staining in the first line of the skin, and in the next lines you can see the immunohistochemistry with Ki67, to evidence the proliferation of the tumor, S100 protein and tyrosinase to identify the tumor formation and the presence of melanocytes. With this monomer cell line A375, this allows to obtain tumors in all conditions tested, but the epidermis stratification got compromised due to the rapid growth of these tumor cells. And it was observed a tendency, as you can see here in the red squares, a tendency to develop larger tumors when the number of monomer cells present in each sample was greater in both type of models. Relatively to the second monomer cell line, G361, we also obtained tumors in all conditions, and we observed an increase in the tumor size with a higher number of monomer cells initially introduced, but only in the model with aggregates, or in the model CA. Also, the increase in tumor size can prevent the correct stratification and differentiation of the epidermis, affecting its thickness. And in both models with these two monomer cell lines, the previous one and this one, tyrosinase show that the development of tumors also affected the organization and the localization of melanocytes. Lastly, SKML1 proved not to be so easy to use in our model. As you can see, only 2 of the 8 conditions tested developed tumors, and only in the model CA, in the model with aggregates. For each sample, the number of tumors was counted and divided into groups, present only in the epidermis with the white bars, and present in the dermis and epidermis with the gray bar. With these two monomer cell lines, A375 and G361, it was observed that with a higher number of monomer cells, the number of tumors was larger, leading to an increase in the number of tumors that proliferate in the epidermis and invade the dermis, and with the A375 in both models, and in G361 only in the model with monomer cells were seeded directly on dermis in the model CD. On the other hand, SKML1, with the SKML1, there was no relationship between the number of monomer cells seeded and the number of tumors formed in each sample. So, to conclude, our cutaneous melanoma model was successfully established. We developed an in vitro melanoma model with melanocytes and 3 different melanoma cell lines, a model with a good differentiation and stratification of the epidermis, and it was developed in a shorter culture time, only in 25 days. Finally, for the next steps, we want to introduce components of the immune system to study the role of immune cells in the tumor microenvironment. We want to use patient cells to personalize the model, and we want to use the model to test anti-tumor drugs. And in the future, this model will provide a useful tool, for example, to characterize the melanoma microenvironment, to study the mechanisms affecting melanoma progression and invasion, to study new therapeutic targets for melanoma, and to personalize the model with the cells of patients in order to find the best therapy for each patient. So, thank you I'd like to thank you to my supervisors, Isabella, Liv and Marta Pous, all the people in our lab and the anatomy service in IPO de Lisboa, and I also to thank you the founding agency of this project. Thank you for your attention. I'm happy to answer your questions, and sorry for this problem with the PowerPoint presentation, and thank you. I don't know if you have questions.
Thank you, Dr. Sarah. Is anyone having any questions? I think there are no questions. Thank you, Doctor, once again for your presence and presentation.
Okay,
Let's move to our
thank
next
you.
presentation. Yep, thank you. Let's move to our next presentation by Dr. Sara Crucian from University of Sassari, Italy. Doctor, are you ready with your presentation?
Yes, I'm here.
Okay, now I'm going to make you as a presenter now. Please share your screen.
Yes. I can share my, okay, maybe now.
Are you able to share your screen?
Just a moment. I have to leave from the meeting and enter again.
Okay. Okay, we'll wait for your. Thank you.
Okay, I'm here. I need the authorization to share my screen.
Yes, I made you as a presenter. Please do share your screen.
Yes. Can you see my presentation?
Yes, I can see your presentation. Can you make it as a full screen? Yeah, perfect. I think you're having low internet connection. Please that so that it won't get any disturb.
Okay, can you see?
Yes, perfect. Please continue with your talk.
Thank you. Good morning, everybody, and thank you for the chance to present the results. I'm a postdoc from University of Sassari, and these results were focused on the control of adipogenic differentiation and inflammation in stem cells isolated from adipose tissue as novel approaches in regenerative medicine. Regenerative medicine identifies the area of research in which we can use stem cells for tissue regeneration, and several diseases as cardiovascular, musculoskeletal, or neurological disease represent a loss of functionality of or aging or tissue and organs. So stem cells can be used as important source to ameliorate this genetic and degenerative disease. Stem cells are in fact dead cells whose fate has not been decided, and are characterized by their ability to self-renew and to maintain tissue homeostasis. So, under specific chemical and physical stimuli, can differentiate into several cell types that compose tissues and organs. In the recent years, several sources of adult stem cells can be identified, including bone marrow, dental pulp, umbilical cord, and in particular, we use adipose-derived stem cells to achieve this goal of regenerative medicine and induce to differentiate into different phenotypes. Adipose-derived stem cells are largely involved in the recent year in therapeutic application in this regenerative medicine, and they can differentiate not only into adipocytes, mature adipocytes, contributing to maintaining tissue homeostasis, but under specific stimuli from the microenvironment, they can differentiate into different cell types. Inside of the stromal vascular fraction of adipose tissue, we can find several cell populations, pericytes, endothelial and progenitor cells, pre-adipocytes, and mesenchymal stem cells in the adipose tissue are a very small number of this population. So we can obtain from the stromal vascular fraction isolated or by lipoaspirates or during mechanical procedures. Adult mesenchymal stem cells can contribute to normal adipose tissue homeostasis, and these cells isolated from stromal vascular fraction can be considered pericytes or perivascular progenitor cells. And as mesenchymal stem cells isolated from other sources, as bone marrow, for example, they have a fibroblast-like morphology and several characteristics equivalent to the other multipotent stem cells isolated from the other tissue. For a long time, adipose tissue is considered as a passive organ dedicated to lipids storage during excessive caloric intake and able to release energy when needed from other organs. But actually, we know that adipose tissue preserve internal organs from each kind of insult and exempt an important endocrine function by secreting several hormones as leptin, adiponectin, and resistin. We can divide adipose tissue into different types, white adipose tissue and brown adipose tissue. White adipose tissue normally store energy as triacylglycerols, while brown adipose tissue dissipates the energy as heat from the acidified metabolism. But between white and brown adipose tissue, we can find a third type of adipose tissue that is called beige or bright. And this type of adipose tissue results from differentiation of precursor cells into beige adipocytes or from the transformation or transdifferentiation of pre-existing white adipocytes, and is classified as beige or bright because has characteristics that are intermediate between white and black and is capable to respond at thermogenic stimuli. The process that transforms white adipose tissue to brown adipose tissue is called browning or hot browning. It is essential in the regulation of whole body metabolism. Normally, during this browning, we observe a decreased inflammation inside of the adipose tissue and a reduced number of pro-inflammatory cytokines with the regulation of the expression of specific heat shock proteins. Normally, the brown adipose tissue present upregulated levels of UCP1 protein, an upregulation in lipogenesis processes, and a reduced oxidative stress. So, every dysregulation in stem cell recruitment and differentiation is linked to a level of chronic low-grade inflammation and macrophage infiltration inside of the tissue. This is typical of the obesity. When we have an excess in lipid storage, we observe a low-grade inflammation inside of the adipose tissue. And obesity is a multifactorial disease that is sometimes associated with metabolic syndrome, type 2 diabetes, cardiovascular disease, and an infiltration of macrophage inside of the adipose tissue. So within this context, we aim at evaluating the role of two important molecules, the metformin and vitamin D, used alone or in combination in modulating inflammation and autophagy of adipose-derived stem cells during adipogenic differentiation, in the attempt to balance white and brown differentiation and counteracting this uncontrolled lipogenesis. We isolated stem cells from adipose tissue sample of adult male and female patients during common surgery procedures, and isolated the cells by mechanical and enzymatic digestion in collagenase solution. Once obtained, the cells were cultured in appropriate flask and then immunomagnetic separated and characterized by flow cytometry to assess the expression of specific surface markers of mesenchymal stem cells. The cells obtained were induced to differentiation in the presence of an adipogenic conditioning medium when we added or vitamin D or metformin or both. This differentiation was performed for a total of 21 days, and at the end of this period, we performed colorimetric assay, immunofluorescence, Western blot, and gene expression analysis. At the end of 21 days, we evaluated the morphology of these cells under an optical microscope, and in this figure, we can see that the cells that were cultured in the presence of vitamin D or metformin of both show a reduced number of mature adipocytes that, as compared to control untreated cells, and the number of these mature adipocytes were significantly reduced if we observe the cells that were cultured in the presence of the only adipogenic conditioning medium. So these molecules show that were capable to reduce the differentiation despite the presence of the adipogenic conditioning medium. So we analyze the principal markers of stemness, OCT, SOX, and NANOG, and we can observe an upregulation of all markers at the end of 21 days, and a reduced expression of the main markers of adipogenic differentiation, that is PPR gamma. PPR gamma is normally upregulated when adipose-derived stem cells become mature adipocytes, but in this case, the presence of these molecules counteract its expression. We then observe the expression of the main marker of adipogenesis, AP2, LPL, and ACO2, and we observe at the end of 21 days of differentiation an upregulation of LPL and ACO2 that are mainly related to the brown adipose tissue to support the storage and the thermogenic activity of these cells, while AP2, that is typical of the white adipose tissue, was significantly upregulated when the cells were cultured in the presence of the only conditioning medium, while the presence of metformin or the vitamin D or both significantly decreased its expression. The ability of these molecules to counteract the appearance of an adipogenic phenotype was confirmed by the other analysis regarding the expression of the two main enzymes that are involved in vitamin D metabolism. We analyzed the expression of cytochrome P27B1 and 3A4, and we observe an upregulation of these enzymes in terms of the gene expression and the protein levels by immunofluorescence at the end of 21 days, especially when the two molecules were added in combination in the differentiation medium. We then perform a colorimetric assay by oil red to evaluate the number of fat droplets inside of the cells at the end of differentiation, and we observe a significantly higher number of these droplets inside of the cells cultured in the presence of the only differentiation medium, while when we use the vitamin D or metformin of both, the number of lipid droplets was significantly decreased. Immunofluorescence analysis also confirm this data, and we analyze the main markers of the late markers of adipogenesis, ESC1 and PAT2, that are mainly involved in the differentiation of white adipose tissue. And we can observe that, especially when the cells were cultured in the presence of the both molecules, the expression of this protein was reduced, while when the cells were cultured in the presence of the differentiation medium alone, its expression was significantly higher. Finally, we analyze another markers of beige adipocyte, that is TMM26, and the levels of UCP1 protein that is related to the thermogenic activity of these cells, and we can observe a significantly upregulation of this protein by the first 7 days of culture until the last 21 days, and an increased expression of TMM26 protein when the cells were cultured in the presence of vitamin D and metformin in combination. So we analyze also the levels of the main cytokines, pro-inflammatory cytokines related to the adipose tissue inflammation and obesity, interleukin-6 and TNF-alpha, and we observe that cells cultured in the presence of the differentiation medium alone show significantly higher levels in the secreted cytokines, while these levels were significantly reduced when the cells were cultured in the presence of vitamin, metformin, or both. As last, we analyzed also the autophagic process that is strictly related to the adipogenesis, and in particular, autophagy is activated during adipose-derived stem cells adipogenesis. Our results show that in the presence of these molecules, there was an inhibition in the autophagic process, and in particular in the level of the last protein involved in the autophagosome formation, LC3B1 and 2, while the cells induced to differentiation in the presence of the differentiation medium alone show the typical the presence of this protein, suggesting us that probably vitamin D and metformin in combination may prevent this inappropriate activation of the cells and obesity-related complication. In conclusion, we can affirm that these molecules were able to counteract the adipogenic differentiation, enhancing vitamin D metabolism and inducing the cells to differentiate into this beige or brown-like phenotype, controlling adipogenesis and the final phenotype of these cells, and can probably suggest us that we can apply in the future these molecules in clinical practice to counteract the uncontrolled activation of these cells and preventing the obesity-related metabolic disorders. So I want to thank my research group in Sassari and our collaborators from University of Sassari and Bologna, and all of you for your attention.
Hi, I have a question.
Yes.
So thank you for your presentation. According to your data, metformin could be used for the treatment of adipose tissue while right now is used for the control of hyperglycemia. But are there any side effects from this molecule in the treatment of patients like this?
Okay, yes, the metformin is currently used as a drug for obesity patients that have type 2 diabetes. We test the same concentration that is actually used in clinical practice, but we test for the first time in combination with vitamin D, because we previously analyzed the effect of vitamin D in modulate the differentiation of stem cells, blocking adipogenesis and inducing osteogenesis. So we need to study the exact mechanism of action of these molecules, because we actually we don't know if there is a direct interaction between metformin and vitamin D, or metformin may act through different signaling pathway.
Okay, thanks.
Thanks, you too.
Any more questions? I think there are no more questions.
Okay.
Thank you, Dr. Sarah, for your presence and presentation.
Thank you too.
Thank you. Let's move to our next presentation by Dr. Michael from Warsaw University of Technology, Poland. Doctor, are you ready with your presentation?
Good evening. Yes, I will try to.
Okay. I made you as a presenter now. Please share your screen.
Okay, could you see my presentation?
Yes, I can see.
Okay, so I think that we can start.
Yes, please
So
continue
thank you
with your
for
talk.
introducing me. I will be today talking about polyglycerol sebacate, a quite new polyester which we developed for specific to electrospinning uses. PGS, polyglycerol sebacate. Could you see the second slide? Yes, so it's everything okay. The polyglycerol sebacate is a quite new polyester. First time it was synthesized in 2002 in Robert Langner's team. And it's projected for tissue engineering. Its biodegradability, biocompatibility, non-toxicity, and what is also important for industry, the reagents are cheap and available, and the production technology is quite simple. All that is due to building this material from glycerol and sebacic acids. So in most of cases, the synthesis is a simple polygate condensation of glycerol and sebacic acid. And we could say that there are two steps of this reaction. In the first step, the prepolymer is generated. It could be branched, but the branching is about up to 40%. And then this material could be cross-linked, and cross-linked material is non-soluble and high molecular weight. In some works, these steps are in one reaction, in one form, but the cross-linked material couldn't be used in electrospinning. So it is a significant difference between prepolymer and polymer. Yes, it's a big difference because prepolymer is near linear structure. In this work, our prepolymer is about less than 20% of branching. It's easy processing material, good solubility, which is must have for electrospinning, but low molecular mass, it could be big disadvantages in some cases. After cross-linking, the polymer is insoluble and hard processing because it's a thermoset material, but it has very good mechanical properties similar to rubber. Sometimes it's called biorubber due to its bioproperties like biocompatibility and biodegradability. So we decide to do specific synthesis for a specific PGS dedicated to electrospinning. We know that this material should be good for tissue engineering, but we don't know if this material could be electrospinning alone without any additives, and we don't know how it could be cross-linked as a non-woven. So in the first tries, the materials which was synthesized like in the other works could not be cross-linked in the low temperature, but high temperature occurring for a long time make from wovens films, because all wovens melt during the cross-linking process. So we have to develop the material which could be cross-linking very fast or in a lower temperature. So we do two types of prepolymer: L-pre-PGS, which is similar to commonly prepared PGS in other works, and we make H-pre-PGS, which is our new material developed specific for electrospinning. This materials has different ending groups. In case of H-pre-PGS, there is a small amount of free hydroxyl groups because this material is near cross-linking. It's not cross-link, it is very good soluble, but it is much more near the critical point of cross-linking, and it could be seen when we conclude that ester number of H-pre-PGS is much higher than ester number of L-pre-PGS. So after creating those two polymers, we try to electrospin them, but we could electrospin this material without any additives. So we decide to electrospinning with PLA. And we try different concentrations, 25%, 15%, and 75% of PLA and PGS. We could electrospinning from hexafluoroisopropanol solution, concentrated 3%. The feed rate was 1.2 milliliter per minute. The voltage was 11 kilovolts, and needle collector distance was 14 centimeters. Collector was a drum, 8 centimeter diameters, and it's rotate with the speed 215 RPM. In all cases, our wovens have a good morphology before cross-linking and after cross-linking, but in the case of H-pre-PGS, this cross-linking was not even in a smaller with lowest temperature, but also with lower time. So here on the graph we have cross-linking time and cross-linking efficiency. Here we have three wovens with L3-PGS type, and here we have 3 wovens with H3-PGS. The wovens have different concentration of PLA and PGS. In case of H3-PGS, we see that about cross-linking was in 130 degrees Celsius, and in case of H3-PGS, as you can see, 80% of cross-linking is about 6 hours. After 24 hours, we have 90% of cross-linking efficiency. In case of L3-PGS, after 24 hours of cross-linking, in the best sample, we have 45–43% of cross-linking efficiency. So because of making H3-PGS, which was synthesis for much higher yield of conversion of functional groups, we do the much faster cross-linking material. So in the end, conclusions are that slight difference in PGS structures can much affect cross-linking, and this is because there was synthesis was different in these cases. In H3-PGS, we remove water in synthesis, and it allows us to get material which are closer to critical point for cross-linking. H3-PGS can be cross-linked under lower temperature than in previous works, because in previous works cross-linking temperature was about 180 Celsius degree. The lowest was 150. In our case, it was 140 Celsius degrees. H3-PGS allows avoiding destructing wovens morphology during cross-linking. Our materials that we manufactured could be interesting for tissue engineering. This work is still in progress. We plan to do cytotoxicity test, mechanical strength test, and biodegradability test. And it's all. Thank you for your attention. Do we have any questions? Could you hear me?
Thank you, Dr. Michael. Is anyone having any questions? I think there are no questions. Thank you, Dr. Michael, for your presence once again.
Thank you.
Thank you. As per the schedule, let's have a lunch break for half an hour. Please stay connected. We'll get back for the afternoon sessions today. Thank you.
This conference will now be recorded.
Hello everyone, can you hear me?
Yes.
Hello. Yeah, okay. Thank you. Sorry for the delay. And let's start with our afternoon session. Our first talk is by Dr. R.N. Chatterjee from University of Kolkata, India. Doctor, are you ready with your presentation?
Yes, I'm just okay. Uh,
Okay, now I'm going to make you as a presenter now. Please share your screen.
Thank you.
Thank you.
The topic of my lecture is developmental defects in the imaginal disc of the superior
So-
cluster
Sorry
simulates
for the
hybrid
inconvenience,
stage.
doctor. We are unable to see your presentation. Please share your screen.
This is that cannot be. I just have to be from the mobile because
Okay.
my computer is not working.
Okay. Okay. I'll display your presentation. Give us a minute.
And just
Okay.
my picture has already gone to your places because it
Yes.
has not.
Yes.
Is it?
Yes. Yes. We are having your presentation. We'll display, and you can announce to change the slides. Okay, so that I can change the slides as per your.
I could not follow it. Can you please repeat it? Hello?
Hello. I'll
Uh,
I'll present your presentation through my screen sharing. Please guide me
uh,
to change the slides. Which slide you want to present? Okay, one by one.
first slide I just want to present
Yes,
because
just give us a moment. Yes, wait, just
uh,
a moment.
I cannot share the. Can you
Mm,
share the slides?
Yes, we are having your presentation. Just to give us a minute, I'll share it.
Okay.
Okay.
Uh, should I read now? No, it's not necessary. Is it? Is it necessary
Yes, you.
for? Hello?
Hello.
Is it necessary to
Yes.
read it that presentation? No. No. Okay. Thank you.
So please guide me so that I can change the slides. Okay.
Okay.
Okay. Thank you. Please continue with your talk.
The title of my lecture is the developmental defects of developmental defect of imaginal disc in the Drosophila melanogaster simulant hybrid males that are caused by the maternal zygotic transition failure. Next slide. Next slide. Next slide. This is the each species has its own history and may have accumulated different modification of genetic system. In absence of gene flow, the genetic distance between population is established, preventing the gene exchange required for isolation as byproduct. Mechanisms preventing the gene exchange between species are not homogeneous, and most of them are thought to be result of genome size variation. The many satellites and T is abundant. T is transposable element abundance varies among the species, and are the major contributor in the genome size. Lethal hybrid incompatibility mainly of two types in prezygotic and postzygotic. Here, Dobzhansky-Mahler model explains how prezygotic reproductive isolation is a major event for speciation in natural population. Next slide. As mentioned, the speciation accompanies reproductive isolation in natural population through the hybrid sterility and lethality. One of the best characterized examples is the hybrid incompatibility gene, one pair of genes, the hybrid male rescue of Drosophila melanogaster, hybrid lethal hybrid rescue in Drosophila similants. Since loss of function mutation on either of the genes can suppress the lethality, and since the two genes suppress the heterochromatic repeats, it is widely accepted that H-humor and LHR are major contributor of hybrid incompatibility between two species of melanogaster similants. The molecular nature of the two genes, LHR encodes HP3 of 319 amino acids. H-humor is also isoform of HP3 and it has a bigger size, but both H-humor and LHR interact physically and localize predominantly in the centromeric region. LHR and H-humor regulate the species-specific satellite DNA of the two species, and LHR protein and MHR protein are required to suppress the transposable elements of the species. Here, the two species has been, the phylogenetic relationships of the two species has been shown, and melanogasters and similants has been separated 0.8 million years ago. Next slide, please. The cross showing that the incompatibility interactions occurs with the maternal cytoplasm, melanogaster X chromosome, and speciation factor HMR and LHR. And if we loss of the LHR factor, it will survive the male. Even if loss of the LHR factor, when we have a attested chromosome process with the simulants, it will also help to the survivability of the female. Next slide. Next slide. Here, earlier, please. Earlier one. These are the polyethylene organization patterns in the lethal hybrid, and these are the male X chromosomes of lethal hybrid. It's the very condensed one, and the hybrid is stored in absence of LHR. And this is the simulant X chromosome which has been already viable. It is key to incorporation pattern of male rescue habit and the viable habit is compatible. These are the female. This is the normal female, and this is the partial hybrid female having X chromosome from the melanogaster species of two exchromosomes of the Madagascar species. And this is the metaphysical 2x chromosome to the Madagascar 1x chromosome of the single species. Here we can show that even if in habit condition, the locus specific transcription is conserved. That means in presence of the arsenic condition and also in presence of synapse condition, and it can be used by red arrows. Next slide. Next slide. Next slide. Here, that is skipping. One of the main example we have shown that earlier slide, back please earlier slide because it has been jumped. Can I have an earlier slide? Earlier slide, can please? No, earlier slide. The next slide. Can I present from that my mobile one? It is convenient one. Can I present from that mobile one?
Yes, doctor. You can present through your mobile also.
Bit better.
Okay.
Okay.
Just a
I'm just
moment. I'll give.
mobile
Okay.
one.
Okay. I made you as a presenter now. Please share your screen.
That mobile, it is shared by the things. Okay. It has
Yes,
been
you can
shared.
share your screen. Yes, you can share. Please click on the share screen button and open your PPT.
Okay, ma'am.
Okay. Yes,
Okay,
we can see
madam.
your screen. Can you please click on the PowerPoint presentation?
Okay, madam. Thank you.
Yes, we can see your presentation. Please continue with your talk.
Okay. Now, I just want to show how the X chromosome organization is regulated. Lessons from the chromosomal rearrangement of inversion PM2 PV industrial Madagascar. PV is the position effect variations. Here you can show that when the chromocenter has been left one, the chromocenter has been diffused. That means the heterochromatin has been the displacement is occur from the chromocenter. Then the organization of the chromosomes has been changed considerably. And if you show, the teeth that stock at the lower temperature level, that means 16 degrees centigrade, it can be observed that the many ectopic pairing has been occurred into the chromosomes within the X chromosomes and also X chromosomes on autosomes also. And here the right one, you can show that the many of the ectopic pairing is of the activity when transposon activity has been taken places into the male. So from that, and when you have a duplication of the X chromosome, a segment of the duplications of an A, B, C, D, and so on, I mean 16A, the duplication of the X chromosome is going on, then the PV phenotype has been modified and considerably. That means the termination of heterochromatin spreading on the X chromosome in different genomic contexts indicates that there is an inherent mechanism for movement of heterochromatin binding protein on the X chromosome. This observation suggested that the chromosomes are not a passive collections of genes. They contain an internal structure that has a role for regulation of X chromosome organization. Within summary, I can show you that the chromosome structure and genome organization. Each chromosome has a centromere and telomere. And here we can show that each chromosome has a complementarized part. One part is the euchromatic region, other part is the heterochromatic region is associated with the centromere. So this heterochromatic region is responsible for the changing the organization pattern of the genome or chromosome for epigenetic inheritance pattern of the chromosomes. The type of developmental defects with the background, we just want to know the type of developmental defects in lethal hybrids. As mentioned in the interactions between the HMR and Escherichia causes hybrid larval lethality. The male larval lethality pathology shows sluggish nature, small imaginal disc in the larvae, and larvae fail to metamorphose. The loss of function of Escherichia and immature is a strong suppressor of larval lethality. That means when adaptive evolution does ultimately lead to a hybrid incompatibility between two species, it is expected that evolutionary forces also shape the regulatory variation on divergence of imprinting of parental genomes during the gametogenesis of species. That means species specific. Therefore, with the mixed genome of hybrid zygote, maternally supplied transcription factor have only permission that epigenetic reprogramming of the modification at early stage of development. This asymmetric reprogramming of the zygotic genome prior to zygotic gene activation may affect the subsequent developmental process in hybrid. Since in absence of developmental problem of the imaginal disc, the comparative analysis of the disc morphogenesis in lethal and hybrid tissues may help us to understand the mechanistic inside the speciation of speciation by HMR and LHR genes in Drosophila. We know that germ cells differentiation into gametes differently in two sexes. Sperms undergo extensive modifications, differentiation, and repetitive pi RNA is associated with the repression of transposable element during the spermatogenesis. On the other hand, during the maturation of oogenesis, the oocyte undergo meiosis 1 and finally meiosis 2 after fertilization. So, this is responsible for the anisogamy. The gametes causes carry the species-specific epigenetic asymmetry between the parental genomes. Upon fertilization, the sperms and eggs unite and produce zygote, which is totipotent, having potential to produce all cells of the body. Species-specific epigenetic memories in both spermatogenesis and oogenesis are heritable to zygote for permissive epigenetic modification in early development. Therefore, it is expected that the switch from the singular function of totipotent cells in hybrids may perturb the massive molecular rewiring process of the execution of early biological process in development. Here, we can show the life cycle of Drosophila. The left two are the pure species, and the red arrows species is the hybrid lethal larvae, and the right one is the only the rescued hybrid. Within the larvae, the imaginal discs are there, and these imaginal discs are formed, are composed of cluster of cells and differentiated into it is responsible for both on development of appendages. And imaginal disc growth and development of the require the three major processes: cell division, cell growth, and cell patterning. Each of the cell process is controlled by specific, highly conserved regulatory pathways. And here we show that wing disc, upper one is the wing disc of pure species, and the left one is the lethal hybrid species, which is very tiny wing disc. And also, Raipanbulu said that the wing disc is very contains the lethal cells. On the other hand, in the rescue hybrid, the disc is grown enough and can proliferate. That means lethality is suppressed if we have one of the species and factor into the genome. So here some other examples we are showing. There is a female genital disc, male genital disc, halter genital disc, and also leg genital disc. In every cases, we can find that when there is a lethal hybrid, the disc was tiny, and it contains high amount of lethal cells. And this has been rescued when one of the species and factor is absent in the hybrid genome. These are the data, and upper one the pure species, and the lower one the red one only the lethal species, and the lower one is a rescued species. And you can clearly reveal that when the incompatibility cause the cell death in the imaginal disc and resulting the death of the larvae. If we have examined that the patterning of the expression patterning genes in the wing, halter, and genital disc, and we can observe that the expression pattern of the patterning genes in the rescued disc is some diffused, or it crossed the complementary organization pattern, a stick complementary organization pattern of the pure species. And here is some example of some wing disc. Upper one is the only the pure species, and lower one you can very diffused one and diluted one, and also complementary organization has been changed. So if we analyze that on a differentiation organization pattern, this left one is the pure species male genitalia, the right one is the left species, hybrid species male genitalia. The development has been asymmetric. This is not only in the case of genitalia, but also wing also have a similar situation. When we make a correlation between the thorax length with the wing length, there is a pure species have a good correlation coefficient. On the other hand, in the hybrid species have a very low correlation coefficient between the wing length and thorax. That means asymmetry of development has occurred in the hybrid species. This is one of the example. Upper one is the pure species, and lower one is the hybrid species. And it can observe that in the hybrid species, the telomeric region has been was defective, and some binding pattern was defective, even if it can restore the hyperactivity phenomenon of the Murex chromosome. With this data, we just want to conclude from the things that the cell death reaction in the imaginary scale in larval in imaginary larval hybrid males are result from divergent lineage of maternal and paternal sets of chromosomes in zygote in presence of species-specific mediator gene LHR plus and HMR plus. This caused upregulation of Ts in the hybrid zygote, resulting the cell death reactions in the Ts. The defects are likely enhanced in abnormally expression of Packard, which is the negative regulator of the GNK signaling pathway at the wound site. Suppression of lethal hybridity in absence of LHR function indicate that the cell death factor in the disc are controlled genetically in incompatible reactions between LHR plus and HMR plus gene. Rescue actions due to absence of LHR indicate that the HMR alone has the potential to rewiring the gene regulatory network in the mixed genome. This specification of complementary organization of the melanogaster segment property genes in the rescue habit might cause asymmetry development of appendages in the hybrid. Since Ts are a major structural component of the eukaryotic genomes, and since Ts has potential to rewire the existing gene regulatory network, we conclude that Ts have biochemical activity also, and Ts not always epigenetically replaced. With this data, how to capitalize this knowledge of the rescue effect of imaginal disc in hybrid disc to address the problem of biomedical impact. Since TEs differ in their frequency and form in different species, cross-species tissue transplantation program may have detrimental consequences on the host. Genome editing tools such as CRISPR-based genome engineering now enable to make any desired change in the genome sequences. This approach can be used to edit or delete the mediator genes, leading to suppression of cell death reaction and of the cross-species transplantation of tissue transplantation experiment. Recently, a significant functional role has been attributed to the T's immune-related process, since 99% T's derived from transcription factor binding sites are specific. Cross-species multiomics analysis would enable better understanding to the species-specific heterochromatic factor effect on T's organism. Thus, CRISPR tools can use the cross-species tissue transplantation experiment in future by editing donor species T regulator or speciation gene or the tissue plantation by tissue engineering experiments. So, I'm thankful for having patience. Regards. Thanks, sir. It's completed.
Thank you, Doctor. Thank you, Dr. Chatterjee. Is anyone having any questions?
Questions. This is the tremendous impact into the medical science, because sometimes we have to transplant because heart or lungs or something, or some tissues are not always available for transplantation. So this can be used from the cross-species transplantation experiments. It is very important thing, because previously it was not known what is the effect and how the the cross-species tissues cannot grow in the host species. This is due to the transposable, species-specific transposable elements and regulatory element present in each species, which is species-specific. So this is one of the very important steps. If we can go progress, it has a very impact in medical science. I can say that.
Thank you, doctor. Is anyone having any questions?
Can you ask any question? Any problem from the, if I can answer, I shall try from your side. Any?
I think there are.
Hello.
Yes. Please continue.
Is there any question from your audience?
I think there are no questions, Dr. Chatterjee. Thank you once again for your presence and presentation.
Thank you, madam, for your patience, and I really thank you for helping me by sharing my data to the August gathering. That's I can say. Thank you for your effort, and I really thank you for the things.
Thank you, doctor.
If any question, in future, you can write me. I shall try my level best to answer from my knowledge, because my knowledge is very limited. I shall try my give the answer at my level.
Okay. Doctor, you can give us your email address in the chat box, so that every speaker having your email address, and if they're having any questions, they'll reach you out there. Okay.
Okay.
Okay. Thank you.
Thank you.
Let's move on to our next presentation by Dr. Sudhakar. Due to some emergency, he was unable to attend today's conference, and he will be joining with us by tomorrow. And our next presentation is by Dr. Orlando from University of Sao Paulo, Brazil. Doctor, are you ready with your presentation?
Yes. Can you hear me?
Yes, I can hear you. I made you as a presenter now. Please share your screen.
I have some trouble with my webcam. In order for this, I send my presentation.
Yes, we are having your presentation.
Yes.
Okay, now I'm going to just a minute.
Yes.
Is it okay?
Yes, this is the presentation.
Okay, please let me know the guidance so that I can change the slides.
Yes. I coordinate with you, okay?
Yes, okay.
Okay. Greetings, everyone. My name is Orlando Algueris Nava, and the presentation today is about PDMS URPWA films, a mini review. The next slide, please. Yes. Polyurethanes are widely used as high-performance adhesives, coatings, and elastomers. However, in order to overwhelm their limitation of resistance to fire, constitute the addition of siloxane-based modifiers like polydimethyl siloxane PDMS. Nonetheless, copolymers based on polyurethane PDMS present low hardness and tenacity. By this reason, these copolymers are reinforced with silicate structures. In this case, in one research group, we use hormosil, organically modified silicates, which acting as reinforcement agent. In turn, these hormosils can act as host matrix for different chemical species like ions, molecules, atoms, and in this case, we select a cluster of molecules like polyoxometalates, in this case represented by phosphotungstic acid, PWA. Thus, in this sequence, it is composed PDMSUR-PWA films, which constitute new hybrid materials which exhibit interesting mechanical and chemical properties. The next slide, please. Can you hear me? Yes.
Yes, we can hear.
Yes. See, yes. In this figure, figure 1, it shows the schematic of the composition of PDMSUR-PWA films containing phosphotungstate. And phosphotungstate is composed by phosphor, tungsten, and oxygen atoms. In this scheme, the urethanes in pink color are linked with the PDMS structure in this scheme in gray color. The hormosils, the organically modified silicates, is inside of the structure of urethanes, and the hormosils network and urethanes structure and PDMS structure constitutes a PDMS-UR matrix. And inside in this matrix, it includes phosphotungstates, and phosphotungstates can include inside of hormosil network in yellow color and inside of PDMs UR matrix. The next slide, please. These films could be prepared with pores exhibiting resistance to the decomposition through of the hydrolysis of urethane bonds. Other properties of these coatings, including wettability, hardness, roughness, which play an important role in the adhesion of fabrics. Nonetheless, more studies are necessary in order to know the biocompatibility and cytotoxicity of these films, and also to discover the possible mechanisms of adhesion of bacteria on the substrates considered. The next slide, please. As was shown in the figure 1, these hybrid materials constitute complex matrices to analyze. It is necessary to develop and examine specific analytical methods in order to better understand the photochromic, antiporosic, and mechanical as chemical properties of this material. For this reason, it is important to identify the distribution of silicates as phototungstates inside this film. In my doctoral thesis, this material was analyzed by a set of methods of analysis and characterization techniques. Among them, I can mention XRF, X-ray fluorescence in different modes, energy dispersive, wavelength dispersive, grazing incidence, and micro X-ray fluorescence. Among other methods of analysis, I can mention laser-induced breakdown spectroscopy, ICP-OS, and neutron activation instrumental. These methods of analysis are illustrated in figure 2. The next slide, please. Yes. This figure, the figure 3, is part of the publication made in 2020, qualitative and semi-quantitative determination of the atomic and molecular tungsten distributions in hybrid hydrourethane polydimethyl siloxane films containing phosphotungstate in applied spectroscopy. In this article, was included this figure about the segregation of tungsten in the matrix of PGMSUR commented in the previous slide, in which the tungsten as in form of PWA in concentrations less than 50% in weight present segregation at surface level, as shown in figure A, and in figure B, when the concentration of PWA is more than 50% in weight, this material present segregation at thickness level. It's this difference that generate the different properties, mechanical properties and chemical properties. These materials present a heterogeneous distribution of tungsten in function of PWA concentration. The next slide, please. Yeah. In the first semester of 2020 was published the qualitative and semi-quantitative determination of tungsten in atomic and molecular form in EDMSUR matrix. In the second semester of 2020 was published the qualitative and semi-quantitative determination of bromine in PDMSUR PWA films. Why? Why? Because these films can be considered as potential candidates for proton exchange membrane fuel cells, PEMSCS. As in these nanomaterials, there is presence of bromine in their preparation, could be given the condition necessary for an improved hydrogen bromine fuel cells and battery system. There is segregation of bromine at surface and throughout the thickness of PDMSUR PWA field, like tungsten, which is function of PWA concentration mainly. These films can constitute systems for the storage supply of energy at macro, micro, molecular, atomic scale. The next slide, please. Can you hear me?
Doctor, can I change the slide?
Yes, please.
Okay, I
Yes.
changed. Thank you.
And the figure 4 of this presentation is depicted the model of bromine distribution for PDMSUR PWA films as function of PWA content. A source was qualitative and semi-quantitative determination of bromine in hybrid hydrourethanes polydimethyl siloxane films containing phosphotungstate in engineering physics. Delegates show the distribution of bromine in function of PWA concentration, and as in the case of tungsten, in function of concentration of this tungsten molecule or cluster of molecules, the bromine presents a segregation at surface and at thickness level, but a difference of tungsten, their segregation is not convergent with tungsten. In many cases are opposite For example, when there is segregation at surface level of tungsten simultaneously, there is segregation at thickness of bromine. And when there is segregation at thickness of tungsten, maybe there is segregation at surface level of bromine. And at right is depicted the possible structure of the different species of bromine and their interaction with PWA clusters. For example, in this figure at right, the background yellow represents the PDMSUR matrix. The green ellipses represent the molecules of water. The light blue tetrahedral structure represents PWA molecules, and blue ellipse represents atomic bromine, and violet ellipse represents molecular bromine. In this structure depicted in this figure represents the analysis of what was published in this article, in which the bromine is present in atomic, molecular, and possible ionic form. The next slide, please. Yes. These new films, which are object of this present mini review, exhibit the following feature and potential application: photochromic and photocatalytic properties exhibiting balance, flexibility, stiffness feature. These fields are potential candidates as proton exchange membrane fuel cells, PEMFCS. The content of bromine and water present in these fields can constitute a hydrogen-bromine fuel cell system due to the action of the phosphoton states in the transport properties of these membranes. These films constitute potential candidates as polymeric biomaterials, which would have many important uses in tissue reconstruction. For the application of these materials are necessary the correlation among structure, composition, and properties, not only for laboratory scale, if not in order to glimpse application at industrial level. For example, anti-corrosion coatings, insulators for thermal and electrical application. Thank you very much. That's it. Reference. Thank you.
Thank you, doctor. Is anyone having any questions? I think there are no questions. Thank you, Dr. Orlando, for your presence and presentation once again. Let's move to our next presentation by Dr. Yan Lim Fan from Global COVID-19 Task Force, USA. Doctor, are you ready with your presentation?
Yes, thank you so much.
Yep, thank you. I made you as a presenter now. Please share your screen.
Yes, one second. Okay, cool.
Thank
You
you.
go ahead. One second. Okay. Okay. 1 second. Give me a second. Applications. Give me 1 second.
Okay.
Okay. Can everyone see this?
Yes, we can see your presentation. Please continue with your talk. Please click on the slideshow button so that we can see your full presentation, full screen.
I did. Can you see it now?
Okay.
Or hello?
Yes, we can see. Yes, we can see. Please continue.
Okay. 1 second. Give me 1 second. I'm having a technical issue. 1 second. Okay. So let me see something. Okay. Screen cancel.
Hello, Dr. Yan.
Yes. Yes. Okay.
Okay. Okay.
So okay. Which screen are you seeing? Are you seeing the full presenter screen or what? I just want to make sure.
We are watching like full. No, slide of third one.
Okay, got it. Yeah, because I have two screens, so let me see swap displays. How about now?
Okay. No, just
Which
a
one are you
minute.
seeing?
Your screen, like we can see that half present. Yes, now it's full presentation, full screen. Please continue with your talk. I think it's got disconnected. Can you please connect it again?
Yeah,
Okay.
1 second.
Mhm.
Okay, let me see. Sorry, options, swap displays. Now, which one can you see?
Just we can see the presentation, COVID-19 clinical presentations.
People can see their screen. Yeah, well, I'm in full screen mode, so I'm not sure why it's not coming through.
Yes, we are. Yes, I think. Please click on the slideshow button.
Let's see. Wait a
Is it
second.
possible?
Let me see. Give me a second. Give me a second. Let me.
Mm-hmm.
Can I stop screen sharing?
Yes, you can stop screen sharing and please click on the screen share again.
Okay, wait a second. So there's the main screen and then there's screen 2. Screen 2 share. Okay. Can you see this slide?
We can see the same COVID-19 clinical presentation.
Can you
I
see a
think fifth
slide?
slide it is.
So you can see it then, right? Okay.
Yeah, but again it got disconnected.
I don't know why, because I can see both things at the same time.
Yes, now we can see the full presentation. Please continue.
Oh my God. Okay.
Presentation overview. Yes.
Okay. So
Is
now,
that is what you're saying? Okay.
yes, now you should be able to see it. 1 second. Let me just. Can I? Okay. Good. 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 Okay. Got it. Okay. Cool. Okay. Sorry. Sorry for the technical issues. So I am Dr. Jan Leifman. I am the former director of the Immunology Division of the Global COVID Task Force. Today, I will be speaking to you about COVID-19, COVID, flu, and cancer, tackling the pandemic with stem cell therapies. The slide presentation overview will be about, first, I'll speak about the clinical presentation of SARS-CoV-2. Then I'll move on to SARS-CoV-2 and COVID flu models. Then I'll speak about COVID-19 cancer model. Then I'll speak about cellular therapies, mesenchymal stem cells, and then I'll speak about the prevention of COVID-19 and COVID flu. So now let's speak about the clinical presentations of SARS-CoV-2. So regarding the clinical presentations of SARS-CoV-2, COVID-19 is a respiratory illness. And this respiratory illness is caused by the novel beta coronavirus SARS-CoV-2, and its rapid spread has caused a global pandemic. The viral infection has broad symptomatic presentations that affect most of the organs within the body. And that is really shown here, where here you have the lungs, the liver, the kidneys, the brain, the eyes, the nose, and then the blood vessels. Now, shown here, you have the most common clinical presentations of COVID-19, where flu-like symptoms tend to predominate. And the symptoms include fever, cough, fatigue, shortness of breath, but then there are also non-specific symptoms, which include sore throat, nasal congestion, headache, diarrhea, nausea, and vomiting, including loss of smell and taste, which is a unique symptom that really precedes the onset of the respiratory symptoms. Now, the spectrum of the disease is very wide, where the condition ranges from asymptomatic to critical disease, and this results in multi-organ failure, whereby the disease can range from asymptomatic to moderate disease to severe pneumonia, and then ultimately to critical illness as well, which is really defined by things like acute respiratory distress syndrome, sepsis, septic shock, and multiple organ failure and organ dysfunction. Now, given the manifestations of COVID-19, the systemic manifestations of severe COVID-19 are varied, and they can present as those shown here, which include acute respiratory distress syndrome, hyperinflammation, acute kidney injury, neurological manifestations, and things like hypercoagulability, which then leads through to thromboembolic disease manifested as pulmonary embolism and or acute stroke. So taking into account this diverse clinical presentation really begs into the big question. Is there a potential model? Is there some kind of means that can really explain all of this? And so herein we discuss the models that our group devised for SARS-CoV-2 and COVID flu. So first I want to bring to your attention our proposed model for SARS-CoV-2. And in this model, SARS-CoV-2 acts via both a direct and indirect mechanism to induce damage. Looking first at the direct pathway, you have the virus bind to H2 receptor on all of the organs shown here. In it, there's invasion, replication, and damage, while also from the lung specifically, you have IL-6 secretion, which then goes into your indirect pathway. And shown here is really the manifestations in all of the organs shown, where in the eyes you have things like conjunctivitis, in the nose you have anosmia, in the heart you have myocardial infarction, atherosclerosis, and hypertension. In the vasculature you have thrombosis. In the kidneys you have acute kidney injury and kidney failure. In the brain you have altered mental status, stroke, and seizures. In the GI tract you have GI discomfort, and in the liver you have things like hepatic injury. Now moving on to your indirect pathway, you have the virus activating antigen-presenting cells, which consist of macrophages, dendritic cells, and monocytes, which secrete IL-6. You have IL-6 coming from your direct pathway. You have your indirect pathway stimulating pulmonary macrophages, which result in increased IL-1, which then downstream results in increased IL-6. And then you have hypoxia promoting increased IL-6 secretion. And IL-10 is supposed to rein in IL-6. But if you think of a seesaw, you have so much more IL-6 that IL-10 is unable to rein it in. And then IL-6 activates downstream pathways. Via cis signaling, it activates lymphocytes, which then turn on pro-inflammatory pathways and downregulate anti-inflammatory pathways. Through trans signaling, you activate endothelial cells, which secrete even more IL-6. And then this hypersecretion of IL-6 in the liver results in increase in acute phase reactants, thrombopoiesis, complement activation, decrease in albumin. In the vasculature, you have promotion of vascular leak, vascular permeability, signal amplification, and also neutrophil recruitment. And then all of this taken into account results in a cytokine release syndrome, resulting in systemic inflammatory injury. So the analogy that I like to use is if you have a person standing in the middle of the road, you have a bus hitting them from the front, which is their indirect pathway, and then the bus hitting them from the back, which is their indirect pathway. And that essentially results and causes the resulting damage. So now during the course of our research, we observed a potential synergism between influenza and SARS-CoV-2. Under this model, simultaneous infection with both viruses will cause a further increase in IL-6 production, which would then yield more widespread systemic inflammation and injury than infection with either virus alone. So in this model, similar to the last slide, I'll briefly walk you through it. So here you have the direct pathway where you have SARS-CoV-2 bind to ACE2 receptor. And here in this box, you have all the organs that it affects. And in each of them, you have invasion, replication, and damage, including the lungs. Now, I put the lungs front and center because that's where the intersection between influenza and SARS-CoV-2 is. Whereas influenza acts and binds via SA glycoproteins to also affect the lungs via the same means. And that results in pulmonary injury. And then from the lungs, you have increased IL-6 secretion, which then goes into your indirect pathway. Now, looking at your indirect pathway, you have SARS-CoV-2 via PAMPs and DAMPs, which are non-specific receptors. Activate antigen-presenting cells, which are macrophages, dendritic cells, and monocytes, which once again lead to increased IL-6. You have IL-6 coming from your direct pathway. You then have hypoxia-inducing IL-6, and then we look at influenza. So influenza also via PAMPs and DAMPs activates pulmonary macrophages, and then it either leads to increase in IL-6 directly or increases IL-1, which then increases IL-6 downstream. Once again, you have so much more IL-6 that if we think of the seesaw model, as I spoke in the last slide, that IL-10 is unable to rein it in. And then you have similar activation of all these downstream pathways, resulting in increased cytokine release syndrome and resulting in inflammatory injury. So now here's the big question. Is it real? Is COVID flu, or as the media like to call it, flurona, real? And the answer is yes. Now, COVID flu is a real thing, and that's what we called it. And when we proposed this model in July, which was published in July of 2020. Now, flurona or COVID flu was evidenced where Israel had been the first case in the world on January 2nd, 2022. And then the United States was not exempt because 3 days later, on January 5th, 2022, the first cases were identified. Now, most of the publications in the literature refer to cytokine response induced by SARS-CoV-2, and they define it as a cytokine storm. But the term was originally coined as a key side effect of CAR T-cell therapy. Now, I want to take this time to really compare and contrast the cytokine storm in CAR T-cell therapy with the upregulated cytokine release syndrome in SARS-CoV-2. So if we look at time-wise, cytokine storm in CAR T-cell therapy is more acute. It occurs within hours, compared to in cytokine release syndrome occurs within days. If we look at clinical features, the cytokine storm in CAR T-cell therapy is defined really as capillary leak, neurotoxicity, and hypotension, as compared to SARS-CoV-2, which is more along the lines of a mild to severe influenza-like symptoms, but late manifestations are unique because you have DIC and you have multi-organ dysfunction. When we look at cytokine levels, the levels are 10 to 100-fold higher in cytokine storm and CAR T-cell therapy as compared to that of SARS-CoV-2. And the cause of death is also vastly different, which is defined as status epilepticus or distributive shock in cytokine storm and CAR T-cell therapy as compared to respiratory failure in SARS-CoV-2. And so the two are vastly different entities with large variability in presentation. So here we'll discuss the models that our group devised, our COVID-19 cancer model. So cancer patients, as we all know at this point, are an increased risk of SARS-CoV-2 infection really due to their immunocompromised status. And we propose really a mechanism by which SARS-CoV-2 causes this multi-organ damage, and this is also via IL-6. Now, hypoxia is also caused by malignancy, and is due to alterations in metabolism. This also results in increased IL-6. So the connection is really between the two models is via hypoxia. And that is really shown here, where I highlight really what the change is, and that is hypoxia. So over time, patients affected by COVID-19 are prone to hypoxia. And hypoxia is also common in malignant tumors where it alters cancer cell metabolism and really contributes to therapeutic resistance in these various cancers. Hypoxia itself was the trigger for increased IL-6 secretion, and then that leads to an upregulation of downstream pathways, cytokine release syndrome, and systemic injury. So similar to the previous models, you have SARS-CoV-2 in your direct pathway binding to ACE2 receptor. You have invasion, replication, and damage in all the organs shown here, except and the lungs are front and center, and then they result in increased IL-6 secretion. In your indirect pathway, you have SARS-CoV-2 via pulmonary macrophages either increase IL-6 directly or increased IL-1 beta, which then downstream increases IL-6. You have SARS-CoV-2 via PAMs and DAMs activate antigen-presenting cells, which results in increased IL-6 secretion. And once again, you have so much, and then you have hypoxia, which is where you have cancer and SARS-CoV-2 interplay. Increase in hypoxia results in increase in IL-6. And once again, just like the previous stories, you have so much more IL-6, IL-10 cannot rein it in, activates downstream pathways, resulting in increased cytokine release syndrome, resulting in increased systemic inflammatory injury. So now let's discuss therapeutic interventions. Currently, there are limited efficacious therapeutic interventions against severe SARS-CoV-2 or COVID flu for that matter. But based on the similarities between the two viruses, mechanisms of SARS-CoV-2 and influenza, we've really proposed the idea of a combined therapy, and where this therapy can control the systemic inflammation induced by both viruses, really as this approach has a higher likelihood of inhibiting pro-inflammatory pathways at multiple points in the indirect pathway. So one promising approach is a cellular therapy that really uses mesenchymal stem cells that has yielded promising preliminary efficacy in COVID-19 patients. And here we show 3 different companies that have used these cells in patients and have all shown similar efficacy. Now, one of these companies is called Pluralista, which did a 28-day follow-up from their early phase clinical trial in 7 patients with multiple organ damage who all made a successful recovery shortly after mesenchymal stem cell treatment. Now, this recovery persisted even at the 28-day mark, and all patients remain COVID-free. The company received US FDA approval for a phase 2 clinical trial for the treatment of 140 patients with severe COVID-19 and acute respiratory distress syndrome. Now, how does this therapy work and why? Why is it so effective? So, let me show you. So mesenchymal stem cells, they act as immunomodulators, where they downregulate the pro-inflammatory pathways and they upregulate the anti-inflammatory pathways. And recent studies have shown that they possess antiviral activity as well. And that's really shown here because when you look at the macrophages, there's two types. There's M2 macrophages, which are responsible for activating anti-inflammatory pathways, and M1 macrophages, which are responsible for activating pro-inflammatory pathways. So what these cells do is they increase M2 macrophages, i.e. favoring anti-inflammatory pathways, and then they decrease M1 macrophages, which decrease pro-inflammatory pathways, i.e. you're favoring anti-inflammation. They decrease dendritic cells, which are responsible for promoting pro-inflammatory cytokines, and that results in a decrease in pro-inflammatory cytokines. Natural killer cells, similar idea, where they promote interferon gamma, which then increases inflammatory pathways. By decreasing NK cells, you decrease pro-inflammation. And similarly, in B and T cells, they're responsible for proliferation, antibody production, cytotoxicity, and by decreasing both of those, you have a decrease in the cytotoxicity, the cytokine pathways, and the pro-inflammatory pathways. And so that is mechanistically how they work and how they're able to do their job. So when we applied this to our proposed model for both SARS-CoV-2 and COVID flu, mesenchymal stem cells should inhibit IL-6 at multiple points within the indirect pathway. And they do this by blocking IL-6 and increasing IL-10, which can then further act to dampen IL-6 expression and decreasing pro-inflammatory pathways and increasing Tregs. And that's shown here. Because everywhere that you see IL-6 and IL-1, it's blocking it simultaneously. Essentially, you're blocking all the big roads, all the small roads, and any roads that promote increased IL-6, and thus you have really no buildup of minimal buildup of IL-6, and minimal ability to activate these downstream pathways to then really cause cytokine release syndrome. And so as you see here, MSCs block IL-6 here, they block IL-1, they block IL-6, they block IL-6, but then they also increase IL-10. And by increasing IL-10, you're now giving IL-10 more of a ability and or capability to halt IL-6 in the pathways downstream. And so when applied to our COVID-19 cancer model, MSC should really inhibit IL-6 at multiple points within the indirect pathway by decreasing IL-1 and increasing IL-10, which can then further act to dampen IL-6 expression and thereby decreasing the pro-inflammatory pathways. And that's really shown here, where similarly to the last figure, you have MSCs blocking IL-1 beta, blocking IL-6, blocking more IL-6, and as a result, you have a decrease in IL-6 net, resulting in a decreased activation to activate the downstream pathways, resulting in decreased cytokine release syndrome and decreased ability to induce systemic injury in cancer patients, allowing them to resume their therapeutic interventions. So shown here is a pictorial schematic of the effects of these cells within the alveoli. Now, mesenchymal stem cells are allogeneic, and thus they can be derived from the bone marrow, adipose, umbilical cord, and placenta. And they're expanded and then reinfused into patients. They act systemically when they reach the lungs. They redirect their immune response that then favors the dampening of inflammation to promote recovery and decrease mortality. And that's really shown here, where you have a pictorial schematic of the sources. And then these MSCs, when they go into the lungs, it results in T cell redirection, a switch from M1, which is pro-inflammatory, to M2, which is anti-inflammatory macrophages, and then promotion of anti-inflammatory cytokines. And these anti-inflammatory cytokines, and so all of this results net in promoting recovery by decreasing inflammation, increasing pulmonary function, and then decreasing mortality. Now, most recently, one of the companies, Mesoblast, has demonstrated promising results from their phase 3 clinical trial and received fast track designation from the FDA. Bonus Bio's Mesencure is another promising mesenchymal stem cell therapy that has demonstrated promising efficacy in phase 2 clinical trials that are now expanding to a phase 3 trial. And really what makes their mesenchymal stem cell product unique is that unlike the others, it uses human fat tissue as its source. Now, the efficacy of these cells was further verified in a double-blind and randomized controlled clinical trial performed by the University of Miami, where this trial was designed to evaluate the safety and explore the efficacy endpoints of allogeneic umbilical cord mesenchymal stem cells in patients with COVID-19 and acute respiratory distress syndrome, whereby you have had 24 patients with COVID-19 acute respiratory distress syndrome were enrolled in this clinical trial. And herein, they took umbilical cord mesenchymal stem cells, expanded them in a bioreactor, and then cryopreserved them until patients were present. Then the patients received 2 IV infusions, 72 hours apart, 100 million cells each time. And here, I just wanted to kind of highlight some of the eligibility criteria, where the inclusion criteria really included patients had to be hospitalized for COVID-19. They had to be aged 18. They had to have a peripheral capillary oxygen saturation of less than 94% at room air, or requiring supplemental oxygen when they were seen by a physician. They had to have a PaO2 to FiO2 ratio of less than 300, and they had to have bilateral infiltrates on frontal chest radiograph on chest CT scan. Now, the primary endpoint of safety was achieved whereby no serious adverse events were noted post-mesenchymal stem cell infusion. And so really this therapy was found to be safe. And when they looked at the effects of MSC therapy, it was associated with improved patient survival. The green curve represents the group treated with MSC therapy, whereas the control represents patients that were treated with various standards of care at the time. And what it showed is after 30 days, 91% of patients survived who received MSC therapy compared to 42% in the control group. By day 30, patients who were treated with MSCs demonstrated improved time to recovery. And then when they looked at the survival by day 30, 100%, yes, 100% of the patients less than age 85 survived. Now, this then continued at the 3-month mark, where MSCs treated with patients did better as compared to the control. They also did a cytokine analysis at day 0, and this is really defined as right before MSC infusion and at day 6. And they observed statistically significant decreases in pro-inflammatory cytokines, chemokines, and growth factors involved in cytokine release syndrome post-mesenchymal stem cell therapy. And that's really shown here, whereby if you look at the green bar graph, you have, compared to day 0 to day 6, a decrease in interferon gamma, a decrease in IL-2, a decrease in IL-5, IL-6, IL-7, TNF-alpha, TNF-beta, and so on and so forth. But cytokine testing was limited because they didn't do a full panel. And so, for example, testing for IL-10, as we had shown in our model to be important, was not done. So in summary, in terms of their trial, they showed that mesenchymal stem cell therapy was found to be safe. No subjects really suffered from serious adverse complications, and therapy was associated with improved patient survival, 91% at 1 month, and 42%, compared to 42% who received standard of care, and time to recovery was significantly improved. Now, I want to also bring to your attention an updated trial that was just published on April 10, 2020, which is a clinical trial from Mount Sinai in New York, assessing the efficacy of Mesoblast, which, to remind you, was really the first company that got approval in the United States, mesenchymal stem cells, to treat 11 mechanically ventilated patients with acute respiratory distress syndrome secondary to COVID-19. And looking at their results, they showed that at the end of their 28-day follow-up period, 10 patients were extubated, with 9 remaining liberated from mechanical ventilation and were discharged from the ICU. The median time to extubation was really 10 days. And this is really all shown in figure A. And then C-reactive protein levels, which correlate with inflammation, significantly decline within 5 days after mesenchymal stem cell infusion. And that's really shown in figure B. So their conclusion was that mesenchymal stem cell demonstrated continued efficacy in treating moderate to severe acute respiratory distress syndrome patients. And this therapy was safe, well tolerated, and resulted in an improvement in clinical outcomes. And so this clinical data is in line with the study that I showed a few minutes ago from the University of Miami. So now the last phase of my talk is really prevention of COVID-19 and COVID flu. We know that flu season is kind of upon us, although it is kind of on the tail end. And COVID flu is a burgeoning pandemic. And in anticipation of this, we really propose a preventative approach that really can be implemented to better prepare for what may come. And the solution is really simple: just get your flu shot. And we suggest that this really is the most effective means of prevention and protection, as the hope is by getting the flu shot, one will be provided with immunity against influenza and thereby decrease synergistic interaction with both viruses. And so by keeping the flu at bay, we can then focus on attaining better control of SARS-CoV-2 and the promising therapies and vaccines currently in clinical trials. And that's really summarized here, where this is clinical. Well, these are recent studies that really looked at, the efficacy of vaccination. And both studies concluded that influenza vaccination is associated with a lower risk of SARS-CoV-2 infection, where retrospective cross-sectional study from July 1st, 2019 to June 30th in 2020, in 280 million adults age 65 or more who had received influenza during this time period compared to those who didn't, showed a 24% reduction in getting COVID-19 and a 28% reduction in the odds of developing severe COVID-19. And similarly, in a meta-analysis that looking at the association between influenza vaccination and SARS-CoV-2 in 16 observational studies in close to 30,000 participants showed that influenza vaccination was associated with a low risk of SARS-CoV-2 with no association with clinical outcomes of mortality, hospitalization, or intensive care. And so it has been shown to be promising. And then the next question, is it safe to administer the flu and COVID-19 vaccines? And really, both studies here show that it is safe, where antibody responses to both vaccines are preserved. Now, in September of 2021, it was announced that Moderna is developing a single vaccine that really combines a booster dose against COVID-19 with its FDA-approved COVID-19 vaccine. Now, this was the headline from January 17, 2022, where given the severity of the burgeoning COVID flu pandemic, Moderna is developing a COVID flu joint booster vaccine with an anticipated release date of late 2023. So, in conclusion, we proposed a cohesive mechanism of action for SARS-CoV-2 and presented the first synergistic paradigm between influenza and SARS-CoV-2 termed COVID flu, along with models and therapeutic interventions for both. Now, looking forward, we see combination therapies being used that can better thwart the virus's heterogeneity, mutational adaptations, and block multiple inflammatory pathways simultaneously and thereby reduce systemic inflammatory damage. Based on our model, we feel that enacting a flu immunization program can really help to provide some form of protection against the synergism of the two viruses until a more effective preventative vaccine and definitive treatment are devised. So, in conclusion, success can only be achieved through great mentorship and working with a great team. I'm very blessed for the mentorship that I'd received from Dr. Sharma and Dr. Chaudhry, and it was a pleasure to work with and learn from everyone on the Global COVID-19 Task Force. And for the cancer COVID-19 study, it was a pleasure to be mentored by Drs. Park and Dr. Sharma and contribute to this important work. Thank you to the great and hardworking team that helped to make this possible. And thank you all for your attention today. Happy to take any questions.
Hi, thank you for your presentation. I have a question. My question is, were these patients treated with autologous mesenchymal stem cells or from a donor? since I wonder whether mesenchymal stem cells are not damaged from coronavirus infection or cancer. So is it better to treat these patients with autologous cells or cells from a donor that is a healthy donor?
So that's a good question. They really haven't done a cross comparison between the two, so I really can't answer it. But I know that right now a lot of the companies are switching to making human fat their main source. And really, because if you think about it, human fat is much easier to procure than, let's say, a placenta or umbilical cord or something to that effect with less logistics. And speaking with several representatives from those companies, they've actually said that they found that their data was better when using human fat as compared to any of the other sources that I had spoken about previously. Thank you.
Okay, thanks.
Thank you, Dr. Young. Is anyone having any questions? I think there are no questions. Let's move to our next presentation by Dr. Mike from William Hospital, USA. Let's wait for a few more minutes to join. Give us a few minutes. We'll contact Dr. Mike and we'll get back. Thank you. Just now we have contacted Dr. Mike. He'll be with us within 2 to 5 minutes. Thank you. Hello. Dr. Mike, are you able to share your screen? Hello. Dr. Mike, please respond. Can you hear me? Am I audible to everyone?
Yes.
Okay, thank you. Dr. Mike, can you hear me?
Hello.
Hello. Yeah,
Okay.
doctor. Okay. Now I'm going to make you as a presenter now. Please share your screen.
Okay.
Mm-hmm.
Just a minute.
Okay.
Just a sec.
Mm-hmm.
It's not allowing me to. Daggone, it's not allowing me to share
Okay.
the screen.
Doctor, we are having two screens of yours. One is mic and one is for mic bridge.
Right.
Which one I have to give the permission to share your screen so that I can give?
Do on my email. No, on my um
Is it
laptop.
possible to share your screen now?
Yeah. Yes.
Okay.
Yes.
Mhm.
Yes.
Yes, I made you as a presenter now. Please share your screen. Please click on the share your screen button, the bottom of
Right.
your GoToMeetings app.
Sure, I'm trying to do it, but it's not allowing me to do it. It keeps
I
go to
already gave
meeting.
you the permission. Yes,
Yeah.
please in the GoToMeetings app,
Go to
bottom of
meeting.
your GoToMeetings app, you'll be having four icons.
Okay.
You need you need to click on the third icon, share button.
Okay.
Then you need to give the access to share your screen. Then you can present your talk.
Alrighty, let me try again. See,
Okay.
because for whatever reason, it's not allowing me to do it. Open.
Please give access.
Yeah, I'm
Yes.
trying.
Okay.
It's
Please open the GoToMeetings app.
Yep, I'm doing that.
Okay.
And it says
Have you seen share icon?
yeah, I do, but for whatever reason, let me see if it's leave meeting, cancel. See, it says go to meeting needs accessibility features. Ah,
Yes, you
fuck.
need to give the access to share your screen.
Right.
Yes, please
Accessibility.
give. Yes, please give the access to the screen.
Huh. Uh, huh. For whatever reason, it's just not allowing me to open it.
Okay, can you share your PPT with us so that we'll display on your behalf and you can present over you
Yeah, let
can.
me do that. Let me do that. I'm gonna
Okay,
send it to you via big files.
okay,
Let's do it that way. Just a minute.
okay. You can send us an email.
Correct.
Is it possible? Yes.
Yep, I'm gonna do that right now.
Okay, we'll check our email.
All right, just a minute.
Okay.
Huh. Here we go. I'm gonna forward this to you. What's the email? Hello? Hello? Oh boy.
Have you sent the mail?
Yeah, I'm trying to get your email. What's your email? What is your email address? Hello?
Just a second. Just a second.
What's a good email? Ah, oh, it's too hard. Yes.
Hello, doctor. It is tissuescience@crgmeetings.com.
What is it?
Tissuescience@crgmeetings.com.
Spell it.
.org.
Spell
Tissuescience
it.
at CRG. TISSUE.
T-I.
SSUE.
Uh-huh. Okay, I got it. All right, so I just sent it to you, and it'll be in a transfer big file, and you can open that up. Yeah. Huh.
Have sent us an email.
Yeah, I just sent it. It should be
Okay.
in. Did you see transfer big file? Just refresh your.
Okay. Just to give us a minute, we'll check with the email and get back. Okay.
Uh-huh. Hmm. Hello? Hmm. Hello? Were you able to get it done?
Yes. So thank you. We received your email
Okay.
and we'll present. Please guide us to share for the changing the slides.
Sure.
Give us a minute. Okay, now I'm going to share. Are you able to see my screen?
Yes, I do.
Okay, please continue with your talk and let me know when can I change the slides. Okay, please guide on that.
Okay, perfect. Today I'm going to be talking about human stem cell based intervertebral disc regeneration for spinal disorders. I apologize, but I'm a busy surgeon, so I was occupied with a case. But thanks for being patient. Next slide. So these are my disclosures. Next slide. So degenerative disc disease is a very common disorder. Its annual incidence, roughly 297 million, resulting in chronic back pain, um, 90% of compensation cost. And in the United States alone, direct and indirect costs for lower back pain management is estimated of 50 to 100 billion annually. So it is a very costly problem, and trying to find cures or treatments for this, particularly in the realm of regenerative medicine, led to our interest in looking at stem cell treatments. Next slide. Next. Next slide, please. So the intervertebral disc is a unique environment. It's comprised of an external annulus and an internal nucleus pulposus. The annulus is much like a ligament. The nucleus pulposus is where the water is held for the disc, and it is a great environment for potential stem cell application. Next slide. So the unique anatomy of the intervertebral disc, in that it's much like a radial tire. There's outer layers of lamina, and then the inside is the nucleus pulposus. It is avascular. It is relatively acellular, so it's a very unique environment. It's innervated on the outside by the sinovertebral nerve plexus, and it is what we have found in our studies. It's an immunoprivileged site, means that you could take tissue, even xenografts, from other sources, and you can implant it into the nucleus pulposus, and they won't be rejected, in part because there's no blood supply to it. So it's a very unique area for potential application of stem cell biology. Next slide. The unique thing about the nucleus pulposus is it is felt to be formed by notochordal cells. These populations of cells disappear at a relatively young age in humans as well as in animals. And you can see from the table there, the notochordal cells, the age of loss of notochordal cells in the rat, age of skeletal maturity 2 months, age of loss of these cells is 12 months. And after that, we start to see the onset of disc degeneration. In the rabbit, 6 months. The horse loses its notochordal cells very quickly at birth, which is why you see a lot of horses, they have a lot of degenerative disease in their disc. In fact, we have studied, or groups have studied degenerative disc in horses, particularly in the neck. because as they lose these notochordal cells, the disc starts to degenerate. In cats, 18 years, and in humans, at roughly 10 years of age. Even though we are skeletally mature at 20, at 10 years of age, we lose these notochordal cells. Now, why are these notochordal cells so important? Well, they are felt to produce the glucosamino glycan matrix, hyaluronic acid matrix. These are molecules that are within the nucleus pulposus that hold water. And as these cells are lost, the amount of matrix or glucosaminoglycan content starts to disappear. And this is where we start to see the disc degenerate, collapse, and dry out. Next slide. So this is an example of what happens when we're young. 70% or younger, and we have a fully viable disc. 70% of the load is on the anterior 2/3 of the disc. 30% is on the posterior aspects of the spine, which are made up of the facet joint there, and also what's called the ligamentum flavum. Next slide. As the disc, nope, go back. One back. There you go. As the disc degenerates and dries out, that load, 70%, is now transferred to the posterior elements of the spine. And this results in hypertrophy of those elements. Next slide. So you can see the green bars are basically in a normal healthy disc. Kilopascals are the forces that are applied in lying down. Obviously, there's not much force when we lie down. And in fact, if you look at a disc on a person in the morning, it's nice and plump and full. But as they stand and sit and particularly bend over, the disc starts to collapse because of increased pressure applied to the disc. And then those other bars, you can see you can see the amount of pressure as the disc degenerates. It no longer functions the way it should. It starts to have less holding force for the vertebral column anteriorly. Next slide. So as this occurs, there's force applied to the posterior elements of the spine, and they hypertrophy. This is a cross-section of an MRI in a patient with severe spinal stenosis. And the joints in the spine are much bigger than they are normally, and the ligamentum flavum is also much thicker than it is normally because of this hypertrophic process that is occurring. Because the disc collapses, it can no longer hold the force of the spine. It's transferred to the posterior part of the spine, namely the joints or the facets and the ligamentum flavum, and they hypertrophy or they get bigger. Next slide. And this is what we believe is happening, is as the force is transferred from the anterior aspect of the vertebral body to the posterior aspect, it initiates a process called mTOR. mTOR is understood to cause hypertrophy in tissue. So we reduce mTOR and we go through atrophy or towards the left. If we increase mTOR, we go through hypertrophy towards the right. Next slide. So now, how can stem cells play a role in potentially reversing the process that I just described? You know, we are in a very unique area of regenerative medicine. All of you are stem cell researchers and probably know a lot about stem cells, probably even more than I do. And it's a fascinating area because the cell, as we know, can come, it can be developed into a lot of different tissues. In fact, all the tissues that are in the human body. Next slide. So what is the clinical evidence on regenerative medicine for intervertebral disc regeneration? This is just one study, and people have looked at this mesenchymal stem cell or platelet-rich plasma or to treat this condition. Next slide. Biologic therapy to enhance intervertebral disc regeneration. There is now this whole area of regenerative medicine using plasma-rich growth factors and taking autologous cells from people's hips and injecting them into the intervertebral disc. Next slide. So does this, treatment really have any efficacy? And this is where we don't really have good studies, prospective randomized, showing that taking a patient's own cells out of their hips and spinning them down and injecting them is really going to help. There are papers like this out there which stem cell delivery via injectable scaffolds is looked at, but the quality is questionable because we haven't really gotten any really good studies proving that these treatments are efficacious. Next slide. This is using autologous mesenchymal stem cells, and the conclusion was it was safe and feasible for disc regeneration, but it's in the early stage, and the problem with injecting cells into a collapsed disc, there's questionable whether it's really going to do anything. Many of these treatments are available to patients. They're out of pocket. They're not supported by the insurance companies because they're still felt to be experimental. And quite frankly, I'm very cynical. I don't know if we're there yet. Next slide. So we started investigating in vitro disc regeneration using stem cells. And, the first part of this is to get a model. Next slide. So we've also looked at the potential of regeneration of the nucleus pulposus using human cells. And the cell source that we found to be extremely viable and pluripotent was the umbilical cord. Next slide. So the first thing that we did was we set out to find a good animal model. We looked in, the pigs and of course, primates, extremely expensive. The rabbit is actually an outstanding model for disc degeneration because all we did is under fluoro, you could see in the top a image there, that's in an animal lab. Under fluoro, we puncture the disc with a spinal needle. And in about 2 weeks, the bottom image, maybe B, it looks really good. You could see that we puncture the disc where those yellow lines are. That's where we puncture the disc. We start to get a degenerated disc, much like we see in the humans. You can see in that lower A image there, that's a human with an annular tear, essentially a puncture in the disc. The disc is getting dark. So simply by puncturing the disc in the rabbit, we were able to show that we could get a model for disc degeneration. Next slide. And this has been shown in other studies using MRI. We used MRI as well to show well-hydrated disc. And as we puncture the disc, sure enough, we can get it to dry out. Next slide. So this is the process by which we do it. We take a portion of the umbilical cord, we harvest the mesenchymal stem cells. In culture, we're able to differentiate these stem cells into nucleus pulposus cells. We then want to know where they go and what they do, so we label them. And then we take to our animal lab, that bottom picture there is me in the animal lab. We punctured the disc, we see it degenerated, and then we inject these nucleus pulposus cells into this degenerated disc that we've confirmed using MRI. Next slide. Next slide. And then we show preoperatively that we stain these cells. We're able to identify them and confirm that they're nucleus pulposus cells. Next. We are able to show that they express proteins or genes that are seen in the nucleus pulposus, SOX, ACAN, COL2, FOXFX. All these are genes that are expressed in nucleus pulposus. So we confirm that they're nucleus pulposus cells. Next slide. And then we inject them into our rabbit, and this basically shows a flow diagram of how we did that. We degenerate the disc, then we inject these cells that we've confirmed are nucleus pulposus cells. Next slide. And then you can see this is an MRI showing a healthy disc. Then we punctured the two discs where the red arrow is, and then we let them degenerate. Then we injected stem cells, and you can see at 6 weeks after cell injection at the green arrow, we regenerated that disc based on our MRI, and you could see the red arrow, which was our control, and it continued to go on to degenerate. Next slide. So then we show that on these cells that we have injected into the degenerated disc, we looked at nucleus pulposus percentage. It was high in the disc injected with the nucleus pulposus differentiated stem cells. Next slide. And then also water content, very similar to control, and glucose aminoglycan content. Now remember, this is the entity that's created by these notochordal cells. So essentially, these stem cells are acting just like the notochordal cells that we see in the natural history of disc embryology and development. Next slide. And then we could look under H&E, and we could see that those cells or those nucleus pulposus discs that we injected with the stem cells, that we see these chondrocytes, very similar to our control disc. Next slide. We also looked at protein expression in these discs, and we found that protein expression in the nucleus pulposus disc, they expressed the proteins that we normally see in the nucleus pulposus. So this came down to saying that not only these cells that we injected, we confirmed that they were expressing nucleus pulposus proteins. Next slide. We also came up with this mechanism based on our findings and the expression, gene expression of a novel mechanism for nucleus pulposus regeneration using human cord blood stem cells. And I would encourage you to go to PubMed if you're interested. You can go to some of our publications. You can see how we harvest these cells, what portion of the umbilical cord we harvest these cells from. They're from the placental cord junction, have the highest pluripotency. And so we're able to show that we're able to regenerate the nucleus pulposus. Next slide. So now we're looking at hydrogel matrix, and I heard one of the earlier speakers talk about this, and this is an interesting area of investigation simply because it provides a more favorable environment for these cells to grow and into the tissue you want them to grow into, in our case, nucleus pulposus. Next slide. So this just shows the growth of and impregnation of the nucleus pulposus cells that we took from the umbilical cord. We're growing them on our hydrogel matrix. We're analyzing them and showing their expression, and that they express better than just plain umbilical cord stem cells in culture or injected into the hydrogel. So the hydrogel matrix seems to enhance these cells' ability to express nucleus pulposus genes. Next slide. This is how we made our hydrogel. I'm not going to go into detail about this. And then we essentially had these hydrogels injected into an ex vivo rabbit model of degenerative disc. And then we subsequently harvested them and investigated them further. Next slide. And you can see here where we did histologic analysis and showed them to be very robust in restoring the integrity of the nucleus pulposus, as well as transcriptional analysis of implants revealed higher expression of nucleus pulposus genes and markers in nucleus pulposus stem cell matrix. So the conclusion is this study demonstrated in an ex vivo model of disc degeneration in the rabbit a more effective way at regenerating the disc. Next slide. Next slide. So this just shows in detail the hydrogel matrices that we use. Next slide. The discs that were harvested ex vivo that we implanted. Next slide. Next slide. And once again, this is what I just showed you. Next slide. Next slide. Once again, showing how effective. So how can we actually restore the degenerated disc? You see on the lower image is an MRI. We have thought to investigate this distractor that is implanted in a patient. It's very similar to the surgery I did today, except it's not a distractor. It's actually a rod that's between two pedicle screws. But in this case, this is a distractor. This is not FDA approved, and we're hoping to do clinical trials, but it's a long process. But this distractor can then be placed and over time restore the disc height. And then the idea is to take these umbilical cord differentiated nucleus pulposus cells and regenerate the nucleus pulposus. So this is one of our goals. Next slide. And I'd like to thank our collaboration. We've done this for many years with some wonderful collaborators from Beaumont and Oakland University, Wayne State, Michigan Head and Spine Institute, Providence Medical Center. Next slide. This is a textbook. I'm a minimally invasive spine surgeon. If you're interested in looking at what I do, the last chapter of this textbook goes into more detail about some of the studies we've done on stem cell and disc regeneration. So if interested, you can look it up. Next slide. Thank you very much. Any questions? Okay.
Thank
Hello?
you, Dr. Mike. Is anyone having any questions?
There's a couple in the chat box I see.
Yes, it's a very informative presentation. Thank you.
Okay. Well, you're very welcome.
Yeah. Thank you,
Okay.
Doctor, once again for your presence and presentation, Dr.
You bet.
Mike.
You bet. You take care. Bye-bye.
Yeah. Bye-bye. By this, we came to end of the day one conference. I would like to thank all the attendees and participants of today's conference. Hope you enjoyed the conference and gained the knowledge from the today's sessions. And I request you all to attend tomorrow's session too. If you have any queries, you can mail to the concern program manager, and they'll get back to the queries you are having. Thank you. Hope to see you in tomorrow's conference too. Thank you once again for your all presence and presentation.
Thank you, Rachel.
Thank you.
Thank
Bye-bye.
you.
Thank you. Bye.
Thank you. Bye-bye.
Thank you. Bye.
Thank you.
Bye, everybody. Bye-bye. Stay healthy.
Bye. You too, doctor. Bye.